Uh, Good morning, everyone. Myself, Sanjana Vattambar.
Good morning, everyone. Myself, Shrikada.
A very warm welcome to the third annual Swalife International Research Conference on Innovation in Drug Discovery. Myself, Sanjana Vattambar.
Good morning.
Swalife International Research Conference on Innovation in Drug Discovery, organized by Swalife Foundation, Swalife Society for Traditional Science and Innovation, and Swalife Biotech Limited. We are delighted to have you all join us virtually on Zoom and YouTube Live.
Yes, indeed, Sanjana. This conference brings together brilliant minds, researchers, and the scientists who are contributing toward the future of drug discovery and innovations in healthcare. Over the next 2 days, we will be hiring from experts in diverse fields.
To begin with, let's inaugurate and mark the start of this knowledge sharing journey. We request everyone to stay tuned as we move ahead with the inaugurated remark and set the stage for insightful discussion.
Thank you to all who are connected with us. Let's smoothly transition into our first session of the day.
Our first keynote session of the day will be delivered by none other than Dr. Praveen Bade. He will be sharing his expertise on the topic personalized herbal formulation in cancer prevention, as well as known cancer continues to be a global health challenge and the concept of the prevention through personalized approach in gaining momentum.
Indeed, Sanjana. Dr. Praveen Bade sir is a respected academician and researcher whose contributions have been instrumental in bridging the gap between traditional herbal wisdom and modern scientific validation. His ongoing research explores how individualized herbal therapies can be designed based on genetic, lifestyle, and the environmental factors of patients. This personalized strategy could significantly reduce the risk of the cancers, improve overall patients' outcomes.
His insights will not only provide us with a deeper understanding of herbal formulation but also open new pathways for collaborative research and innovation in preventive oncology. We are truly honored to have him with us today.
So, without further delay, let us extend a very warm virtual welcome to Dr. Praveen Bade sir, as takes us through the enlightening talk on personalized herbal formulations in the cancer prevention. Sir, the platform is yours.
Thank you, Shri and Sanjana, for the introduction. So just 1 second, I will just share my PPTs. Okay, so thank you, both of you, and it is very nice to be back. This is our third year of Swalife International Conference, where we try to bring different speakers and also help some of the researchers who want to actually present their work as part of the conference. So to begin with, I will be talking about personalized herbal formulation for cancer prevention. That is what we are developing. That is where we are going. And that's where the Swalife Biotech is working on. But today's session is more about how we are actually moving towards the personalized herbal formulation, and mostly our vision and mission when we started the company in 2017 was to reduce the burden on the cancer patient, and also reduce the or prevent the cancer patients to getting into critical issues with the cancer. We have moved a lot from 2017, where we were working on small molecules of the plant, but simultaneously with the upgradation of the field in AI, we have moved a lot now with a lot of things, and that's what I'm going to share, that how the journey was is going on, and how we are moving towards the personalize herbal formulations. So just to give you the overview that what I'm going to talk about, I will discuss little bit about Swalife Biotech, not going much in deeper, but will introduce some of the things which have been going on. Then the framework which we are using to personalize those herbal medicines, which will be utilized by the patients. Then the third will be the case study um, of the product which we are working on recently. And then how we actually moved utilizing the framework from lab to the clinic, and then what impact and vision we have got. That will be the end of the sessions, and I will be open for any questions. So in the introduction of Swalife Biotech, I will be discussing about different the units which we have got, simultaneously what ecosystem we have developed. So Swalife Biotech believe in collaboration, and the academic, the industry with whom we have a tie-up, they knows that how collaboration is important, how interdisciplinary research is important, and why there is a need of this collaboration and interdisciplinary research. So that way we can actually push the boundaries and have a scientific discovery. So just to give you an overview, when we look at the approach of the Swalife Biotech, which has now, majorly shift due to AI into a personalized approach, where now the well-being has to be in the personalized form, where we are using the traditional herbal remedies, utilizing the digital technologies, so that way we can actually customize the formulation for the the chronic diseases such as cancer, where the prevention or moving into a critical cancer stage can be prevented. So there is a personalized care where which will be taking control of your health, utilizing the holistic well-being approach. So that's where the Swalife Biotech Limited is moving, using the unique path to the wellness. We know that just one size is not going to fit to all. So we go beyond to that, and we are actually making sure that we utilize the synergetic effect of the compound, the secondary metabolites from the plant, so that way it can actually have a unique personalized activity for the prevention of the chronic diseases. So we make sure that we are sourcing all those small molecules from the herbs which are actually from the trusted suppliers. So that way the safety and efficacy will not be an issue. So we do give priority to the safety, and which is very important. So in case of our product which are in the market also, we actually have a QR set, so that way actually the people can actually report any adverse effect or side effect they have when they are taking different medication with the herbs which is there in our product. So when we look at the herbs from Swalife point of view, the herbs are very powerful in sense that they are actually can be multi-targeted. So the one single compound is actually targeting different proteins which have been playing important role in the cancer, but simultaneously different compounds are there in the herbal. Based on the extraction, they are targeting different protein. So there is a multi-target action happening. Then also the cellular protection, like role of an antioxidant, which is actually helping in the DNA repair mechanism. In addition to that, due to the natural compound, there is a reduction in the side effects. So there is a better tolerance profile there, and it is actually moving into the precision targeting, where you can actually make sure that the small molecules which are present in the plant can go and hit those protein, those target which are playing important role in the disease. So it is actually having unique advantages in the prevention due to its multi-targeting ability, and it is going to help in the providing the cellular protection. So due to all this powerful nature of the herbal compound, we are more focusing on the herbal compound. When we look at the disease at the moment from the cancer point of view, there is a global challenge here with the cancer. About nearly 9 million new cases annually have been reported, where the annual treatment is going to about 150 billion dollars. So due to these figures, there is a global burden of the cancer, but simultaneously, the one major issue is that drug resistance rate is very high. So there is about 50% of the drug resistance rate is there for the patient who is been treated at the moment. So if you look at the numbers of cases and the resistance, the number is actually very high in the form of the patients, and there is a major global challenge which is due to this rise in the incidence and also due to the treatment resistance. So there is a high demand for innovation into a preventive strategy. So in this case, we are talking about the cancer, but when we look at the evolution of the people, there is a for other chronic disease also there is a need of this innovative preventive strategies to be developed and can be put in place, so that way the burden of the disease can be reduced. It can be prevented. It can also help when there is a resistance to the drug. So here we actually have developed a R&D ecosystem. I'm mentioning a couple of our divisions, or five actually divisions of Swalife Biotech. We are actually in Swalife Biotech, we are doing research till marketing and sales. So we actually a complete business where we do discovery and we also bring the product in the market. So we have got pure herb division where we are making sure that we are utilizing herb for the preventive purpose. But in addition to that, we have nanopore technology where we actually work on nano emulsion. So we have got about 4 patents published on different nano emulsion systems, which actually help in the increasing the viability of the substances, the secondary metabolites from the plant. So that can be utilized for the target delivery system. Then third division, that is the image expert AI, where we are developing different tools in AI which can be utilized to analyze the preclinical data, but simultaneously the clinical data which can be utilized for taking further decisions. Then the fourth one is Doctor Insight platform. This is a platform where we actually utilizing the doctor's insight in addition to the AI tools which we have developed, and simultaneously it is parallel for the telemedicine. So doctors will be counseling patients, but when they are actually making diagnosing the disease, they are utilizing the AI tool, but simultaneously their own expertise will be taken care with this platform. So it is not a AI tool, but it is a merging of the data from the clinical, which has been utilized by the doctor simultaneously by the AI. And the fifth is the AI tool suite, which I will be showing you that what tools we actually got. So we got from initial in silico study till for pharmacovigilance regulatory affairs, we have developed different tools which actually help in the utilization in different areas of the drug discovery and in the pharma field. So just to go with these examples, I will be sharing some of the example on some of the divisions which are there in Swalife Biotech. So first one, we will go through the first division, that is the pure herb division. So we have got five product under this category. We have actually increased now different products. They will be coming in the market. With these five product, we have done preclinical studies on breast cancer and oral cancer. We have done chemopreventive studies on these five product. Simultaneously, the clinical work is going on. But I will give you an example that how we actually utilize, um, with one example, the preclinical studies. So just to give you an example that, we have done evolution of the chemopreventive mechanism of our pure herb Ocimum tenoflorum, that is the Tulsi plant extract, on the Sprock Dolly rat, where we have used DMBA to induce the oral cancer. So this is the pure herb product of Swalife. So just to give you a background, that in oral cancer, malignant tumor is developed in different tissues of the oral, the like lips, tongue, floor of the mouth. There are total eight sites where the malignant tumor can be developed. But when we look, the most common form is the aqueous cell carcinoma, which is basically originating from the epithelial cell of the oral mucosa. For to treat this disease, the oral cancer, there are chemotherapy drug already available, but yes, there is a need for other therapeutic drug as well. But where we targeted, we actually looked at the chemotherapy induced muscle fatigue. Muscle fatigue here. So when we look at the chemotherapy, mostly muscle fatigue is the common side effect which patients are experiencing. But it's not only the muscle fatigue, it is also making the muscle weakness. And due to this weakness, what is happening, the patient quality of the life while the treatment is going is actually impacting, and there is a the patient is actually having issues with the muscle weakness, due to which there is a reduce in the muscle strength. And due to all these exhaustions and the fatigueness, what is the factors which are actually contributing to this is the on the patient quality of life. And this quality of life is been have issues, and those issues are due to the um, inflammation, oxidative stress, or due to the direct muscle damage caused due to all these chemotherapeutic agent. So when we are looking at this, we are actually looking from two point of view. We want to actually make sure that Tulsi on its own is also effective, but simultaneously when it is given with the chemotherapeutic drug, it is also helping to reduce the side effect. In this case, we have focused on the muscle fatigueness. So we utilize an animal model, that was the Sprague Dolly rat. 30 rats were taken, which were about 6 to 8 weeks old. The protocol was total 3 months, about 8 to 10 weeks. When you actually give the inducing drug, it take 8 to 10 weeks for a animal to develop the cancer. In this case, we were actually looking both. We were looking at the chemo prevention. So the herbs were started from the beginning. And simultaneously, we also continuing it after 10 weeks as well. So it was a completely about 12 to 14 weeks of the study. So the groups were divided into five. Control was where only the vehicle was given. Then the group two, where the inducing agent was given. In group three, the standard drug, that is 5-fluorouracil, was given with the inducing agent. In group four, it was the extract with the DMBA. So the concentration was calculated based on the dose which we have prepared the capsules. So group 4, 55 milligram per kilo of the extract was given to the Sorry. So the tulsi extract was given about 55 milligram per kilo. And the fifth group, as I said, that we were also looking at the chemo side effects. So it was given in addition to the standard drug. So the six animals were utilized for that purpose. So majorly when we are looking, uh, so majorly when we are doing the animal studies, we are looking in case of the cancer, different targets, the proteins. But because we were looking at the side effect and we wanted to make sure that the fatigueness is targeted, we also utilize a behavior study where a rotarod was utilized to understand that how the animal can actually utilize their strength of the body while they are placed on the rod. So at the beginning of the protocol, all animals were put on the rod, and the timing was increased to 300 seconds, and we were looking at the fall of the animal from the rod. There were multiple trials done to understand the reliability of the data. And as I told you, we look at the latency to fall of the animal and the total time the animal actually spent on the rod during this multiple trial. So the numbers of time the animals fall we can be counted, and based on that it can be analyzed. So I will show you the graph. So beginning of the protocol and end of the protocol, we have actually done the study and looked at the results. In addition to that, as I told you, mostly the proteins will be targeted. So there are different assays done in between with the blood, which I have not actually included here. Majorly, I am talking about the behavior study as an example and as the protein that we have targeted. So then the histopath, after the sacrification of the animal, the histopath was done where the tissue was fixed, and then it was stained with the hematoxylin and eosin to understand the different effect of the tulsi and the standard drug. So when we started with the protocol, before starting the protocol, we actually utilized network pharmacology to get some of the targets. In this case, we have actually focused on the common target, which were from oral cancer, from the chemotherapy-induced muscle fatigueness, and from the Tulsi. So total about 1,248 targets were utilized, which were then understand their role in different pathway utilizing the KEGG analysis and gene ontology. Based on their role in different pathways, we actually develop a network where we focus on the protein-protein interaction of those protein in different pathways. And based on the small molecules from Tulsi, we actually have designed a network of the compound and the target utilizing the cytoscape. After doing all these assays, we went for the in silico studies to understand its binding affinity with different proteins. So majorly, we have focused on the 5 proteins which were actually isolated and interpreted by doing different network pharmacology, that is gene ontology, protein-protein analysis, and the cytoscape. We focused on the first 5, and out of that first 5, I'm going to show you the result of one of the protein. But in silico data was very promising with different targets, and we will be working on those different targets in the ongoing project. So in this case, we have identified different targets and the binding affinity with the molecules. But in addition to that, we also focused on the in vitro and the in vivo studies to make sure that the target which we have identified in network pharmacology can be taken further in the clinical studies. So here we actually done a high throughput screening with the DPPHSA, that is 2,2-diphenyl 1-picorodil assay, where we are looking at different concentration of the tulsi extract to understand that where the free radical scavenging activity or the percentage inhibition of the Tulsi extract is high, where we have observed that at the higher concentration, that is 200 or 500, we have actually seen there is an increase in inhibition of the free radicals. Now, this is a in vitro assay to make sure that it can replicate into the in vivo studies. So we have also focused on the antioxidant parameters in the animal tissue, where we have looked at the GCH levels, and we have observed as the standard is also showing increase in the GSH levels. That is the natural antioxidant in the body. Simultaneously, Tulsi and the combination with Tulsi with the standard drug has also shown significant result when we looked at the control. It was also observed with the SOD levels as well, that the combination with the standard is actually is useful when we compare with the control. The MDA levels, when we also looked at the MDA levels, the MDA levels which are very high in the disease has shown significant result when we compare with the Tulsi extract and also Tulsi extract with the standard drug. Majorly when we looked at the result, the synergetic effect of the Tulsi extract with the 5-fluorouracil is more promising. Yes, Tulsi extract on its own is also effective antioxidant, but when we looked at the combination, it is more promising results. So then we moved on like simultaneously. This is after sacrification, but in the meantime also we have collected the blood and we have looked at different parameters. The behavior results, as I mentioned earlier when the protocol was discussed, we actually done the rotor test at the beginning of the protocol, where we have actually seen that there is no significant effect. So all the rat was showing nearly the similar effect on the rotor rod instrument. But when we compare it after 3 months, we have observed that the Tulsi and Tulsi with 5-fluorouracil is actually showing promising results. Now, when we actually, when we look at this from the chemotherapeutic side effect point of view, the Tulsi, when given with fluorouracil, is actually showing good results, and it has been very effective when it is given after the 5-fluorouracil. The histopathic results of the tissue was utilized to understand that what effect it has been showing. So the D group and E group is the major one which we will be focusing. The C is the standard image. So C has also shown that it has actually restored the epithelial structure, and there is an intact layer with minimum side of displaced layer. But simultaneously, when we look at the Tulsi and DMBA group, it has shown that there is a protective effect in the treatment, and it has also reduced the dysplastic changes which have been there when we'll compare with the B group, that is the disease group. Simultaneously in E, as I already mentioned, that the combination of 5-fluoro with DMBA actually have shown quite a good results in all the assays. Simultaneously, the one which I have not shown here also, which were done on the blood, have also given us some of the promising results. So it actually also indicates that there is a therapeutic response in combination with 5-fluorouracil. So the histopath, the behavior studies results are promising. They have actually correlate with the network pharmacology and in silico studies. So based on this, we actually focused on different proteins. Here I am showing you one of the protein which we have done ELISA with, but all the product have actually been tested with different proteins which has been identified in network pharmacology. So we actually utilize TNF alpha, one of the pro-inflammatory cytokines, to check in the tissue, which was increased. So basically, in the disease, there is an increase in the TNF alpha levels. But when you actually give the standard or Tulsi, there is a reduction in the TNF level, and those reduction is non-significant when you compare the different standard and the Tulsi with 5-fluorouracil, but they are highly significant when they are compared with the disease group. So based on this, that we can see there is a good activity in the collaboration, in the synergetic effect, but simultaneously individually as well. So this result actually help us to understand that the pure herb actually giving us lot of benefit when they are compare with the different issues. So moving on to the second division, that is the nanocore, which actually help to increase the viability of our product. So maximum time what is happening, the non-polar compounds, when they are actually showing very promising results, they actually have different viability, and most of the time it is poor viability. So we for to handle that issue, we actually moved into the nano area. And nano, when we came across this viability issue, we went for different nanoparticles, but we wanted to put them into formulation. So when we actually decided to go for different formulation, we actually found that the nano emulsion system is having more promising stability and viability. So we went for that, which helped to reduce the limitation of absorption and viability. Simultaneously, the nano emulsion is actually helping by reducing the size and stabilizing the formulation. So it is actually improving the viability. So it actually is morely about a target-based precision advanced cancer care, where we are focusing using the nano formulation for the treatment purpose. And for that purpose, we actually moved into we have already patented those nano emulsions, and we are moving into the clinical side of that nano emulsion. So this is the second division, where, as I told you, that different plant small molecules has been already put into nano emulsions, and the patents are already been published, and some are in the process of the publication. Now, the third division which I want to talk here is the image expert, which we are mostly now focusing on due to the increase in the LLM tools now which are available and which can be utilized to reduce the phase from the lab to the clinic. So we are utilizing the images, the high resolution images from the databases simultaneously from our work as well, which can be helpful to identify the different patterns. So we are utilizing the AI tool to analyze them, and we are generating those results which can be utilized to train the tools, the machine learning tools. So one of the tool which I'm mentioning here, we just recently got to design patent for that, is the utilizing the isolated chicken eye images and automating it with the AI tools, where you can actually identify if the compound or if the formulation is actually creating any damage, toxicity on the ex vivo tissue, that is the isolated chicken eye. So we got that design patent. So in not only with the chicken eye, we actually have a tool for histopath analysis. Specifically, we have developed a histopath analysis tool for breast cancer, and we are in process of developing the similar type for the oral cancer and the chemopreventive activity as well, to assist the herbal compound in that. The next one, which I mentioned earlier, that is the doctor insight. Here, what we are trying to do is that we are utilizing the expertise of the oncologist, so that way we can actually, develop a preventive medicine. But it's not only from the point of view of the expert, but simultaneously, we are collecting information from the real world evidences, so that with expert network, we will be developing a valid protocol which can utilize for the personalized formulation. So it is going to bridge the research and clinic practice gap which is there when we actually bringing the product into the market, and to utilizing the expert knowledge, that is the collaboration platform where the physicians will be involved. So now also, so this is the the MVP of the that product, where doctors will be registered, and it will be utilized similarly where telemedicine apps are there. But majorly here, we are making sure that we will be including the AI functionality which can be utilized by the doctor, and their own expertise can be utilized when they are diagnosing and when they are counseling the patient. Now, another the product which comes under Swalife Health Tech is the AI tool suite, where we will be working on a platform which is going to utilize network pharmacology, which will help us to map the complex interaction between different compounds and the targets, so that way we can actually focus on the disease pathway. In addition to that, the major issue of the literature, which need to be actually, review need to be done when you are actually working on some of the drugs. So automatic extraction of different insights from those literature can be done, and a database can be developed. In addition to that, we actually have a generative AI model where it can predict the formulation outcome, so that we can optimize it for the further treatment. So this is the dashboard of the tools which we have already developed, and uh, can be utilized from the basic research till the clinical studies, till the doctors using for diagnosing. So in this case, there are a couple of diagnosing tools. One is with the skin, and another one is with the eyes. So if you no, most of the time when you actually go to the doctor, they actually check your eyes. So there are major of the time the chronic diseases at early symptoms can be recognized with the eyes. And one of the tool that we have developed, that is the eye disease diagnosing, it says here, but it can be utilized for for other diseases like Parkinson, Alzheimer's can also be at the initial point can be diagnosed with the images of the eye. So this is the dashboard which can be which we are using for Swalife Biotech product, but which can be utilized for other companies for their drug discovery and their research product development. So these are some of the the areas where we are working, and we are developing our formulations and our product simultaneously, moving like giving the services into the product development. From here, we are now personalizing the herbal formulation for cancer prevention. So there is a personalized framework that we have developed, and it is actually a 4-layer approach where an individual herbal formulation can be put into the personalized formulation for the cancer prevention. So in this framework, the first one is the evidence foundation, where we are utilizing AI analysis, network pharmacology, and evidence integration. So the foundation here is to combine the AI with network pharmacology and establish the scientific evidence for that formulation. So when you are saying AI analysis, we are utilizing the machine learning algorithm to analyze the vast information which is available in different databases. So that way we can have a combination of drugs or molecules, and that can be utilized for the further as a formulation for the treatment. So we are using AI analysis. Then with the system biologic, as I already shown in the example of the one of the preclinical studies, we utilize there this anatopharmacology approach where we have taken compounds from tulsi, targets from that oral cancer, and we have mapped with different pathways. So to understand that what targets have been actually hit by those molecules, and based on that, then you can actually go for a evidence integration where computational and experimental evidences can be utilized and also designed to study with that formulation. So this is the first framework which we are utilizing of AI and network pharmacology to move into the evidence scientific robust scientific evidence methodology. In the second one, we actually have a AI recommendation engine. So I'm just going to show you one of the recommendation system where we are claiming as an antioxidant, but simultaneously we have already proved that into the in vitro, in vivo, and also in the clinical. But we have that system already there, where you are putting the data of the patient, genetics, lifestyle, or health history, and that will be proceed by a machine learning tools. So we will be utilizing the pattern from that, and then the formulation can be recommended and the dose protocol way. Now this AI recommendation engine is purposefully developed for the doctors, so they with their expertise and this recommendation can actually be more personalized to patient by patient based on the data which is there available for that patient. So it is going to actually process the individual patient data, so that way you can actually generate a personalized formulation and the recommendation based on any specific risk if the patient is having due to their health condition or due to their profile. So here I'm showing you that this is the antioxidant targeting recommendation engine, where you can actually utilize the patient context, basic information. Then if they are taking any medication, they can actually put here, and the biomarkers based on their blood test information will be putting, and then we will get the recommendation after running that AI tool, the recommendation AI tool. So this the purpose of this tool is for the doctors. So when they're actually counseling the patient, they will be doing both the things. Now the third one is the clinical insight. Now once the recommendation we got from the AI tool, the doctor need to validate it, and that will be helping the doctor to understand simultaneously their own expertise will be utilized. In addition to this, the literature information will be also integrated. So any latest research or evidences which are available will be also there, and also the real world outcome data which will help into the recommendation algorithm. So this layer is going to give the clinical insight from the AI generated recommendation, and also from the doctor's expertise. The clinical best practices are there. In addition to that, literature with the real world clinical data is going to help to understand the formulation which need to be developed for that person. And the fourth one, where the product will be then developed due to all these things. The product will be designed and it will be optimized based on the information, which can be a normal formulation, or it can be then based on the molecules. It can be a nano emulsion in this case, which will be then tested and standardized for the quality purpose, and then the personalized formulation will be delivered to the patient. So majorly the chronic diseases such as cancer or any other diseases need to be gone through all these four layers, where personalized formulation will be developed. So transfer of this validated recommendation from AI and from the doctor into the high quality viable product can be formulated, and also nanotechnology can be utilized to develop this product. So it is a basically a integration of all these four layer where evidence is collected, then clinical insights are being produced, then personalized layout is being created, which then been put into the product development. So due to all these, the personalized formulation is backed by the robust evidences, which are validated by the clinical expert, and then it is delivered by the advanced manufacturing process, which can then create a personalized formulation. So that's what we are doing, utilizing all this technology and moving into the personalized herbal formulation. So just to give you one of the case study here of a formulation of a treatment which we are developing for the prevention of the breast cancer. So majorly there are three ingredients which are being included here. Selecting active compounds where we in this example there are two small molecule, that is curcumin and naringin. In addition to that, we have got wheatgrass extract, which is actually useful in antioxidant and DNA protection. So curcumin role is for anti-inflammation, and it is also utilized as a apoptosis inducing compound, where naringin is a flavonoid from the citrus fruit, which is fruit mainly for the anti-proliferative properties. So utilizing this from the literature, we have actually got this. And we have selected those bioactive compound which actually already have shown a proven efficacy. In case of the wheatgrass extract, it is already in our database, which where we actually have already developed that product. So now in this, there will be a combination of curcumin, naringin, and wheatgrass extract, because they are focusing on different targets. So then there is a network target analysis, and we have identified these three major pathways which they have been hit by this compound, and they are helping in the prevention of the breast cancer. So BRCA1 pathway, which is majorly the pathway in the breast cancer, then P53, which is the genome protein for cell cycle control, and NFKB inhibition, that is the inhibitory in inflammation pathway, which is suppression and cancer prevention. It is very important in that. So these three pathways are majorly hit by these three ingredient, that is curcumin, naringin, and wheatgrass extract. So when we look at the expected therapeutic outcome, that is oxidative stress, which is actually neutralizing the free radicals and making sure that it is preventing the cellular damage. But in addition to that, cellular repair mechanism and genomic stability is being given by the naringin, and apoptosis induction, that is from the curcumin. So oxidative stress is taken care by wheatgrass extract, apoptosis induction by curcumin, and enhancing the DNA repair by the naringin. So these are the expected therapeutic outcome from this combination of the formulation. But it is majorly the synergetic effect which they are giving and the interaction which is happening. So if you look at the enhancement of the viability, instead of going for the nano, we can actually utilize naringin, which is going to improve the curcumin absorption, and it is going to extend its half-life in the body. So it is going to maximize the therapeutic impact. So just combining naringin with curcumin, it is going to enhance the bioavailability, and simultaneously each compound is actually targeting different aspect of cancer prevention. Due to this synergetic compound interaction, there is a the treatment can actually focus on different pathways and can be personalized for the specific patient. So combining this is actually creating that synergetic effect, and it is going to give the activity which can be exceeded by individual compound. So now the personalized factors will be taken into consideration, where the individual patient characteristics are influencing any formulation or dosing of the the drug. So majorly when you look, genetic polymorphism need to be considered when you are working on the dosing and the composition, also the hormonal status, and also the lifestyle factors. So when you are actually personalizing that formulation, it is majorly these three areas need to be considered when you putting the compounds together. And it has been clinically evidenced that about 73% of the risk has been reduced when the curcumin has been utilized. So there is a reduction in the breast cancer risk by 73%. When you look at the narrowing efficacy, it has reduced the 45% reduction in the cancer cell proliferation, and wheatgrass on its own actually improve the DNA repair capacity. Now, it is very important to understand that the balance need to be happen when you are producing a formulation for chemo prevention, because here the wheatgrass is repairing the DNA, but actually the DNA repair is going to help the cancer cells to continuously grow. But that effect is going to be taken care by curcumin and naringenin, and there where actually the combination is helping. So you cannot claim that a compound which is improving the DNA repair can be useful in the treatment of the cancer. Why? Because the cancer cells are continuously growing, and the one of the mechanism is which is very important is that it has to kill the cancer cells. If there is DNA repair happening, the cancer cells will continuously grow. So that's the reason there is a combination due to which the synergetic effect has been very good, and we have observed this. So this is our one of the product which will be coming into the market. So it is from the lab, now it is moving into the clinic. So translating this research into real world for the prevention is a one of the solution which we are developing utilizing the four personalized layout framework which we have developed. Now in this lab to the clinic, the preclinical evidences are important. So that's what we actually have got. Then it is important to have clinical trial innovation. So having a decentralized clinical trial is going to help and going to fast the transition of these preclinical evidences into the market. So decentralized clinical trial innovations is there. Then integrating the doctors platform is going to help us to get the expert views in addition to the AI, which the recommendation model which we have developed. In addition to that, we are actually empowering the patient through education. So it is important that the people, the patients, knows that how actually things are working when they are taking something. It is recommended by the doctor, or when in case of the herbal, sometime you are taking yourself. It is important to understand what is there in the product and how it works. Then the quality and regulatory compliance are taken care to make sure that the quality ingredients are taken and it is as per the regulatory compliance of that country or for that country. So it is a real world evidences are collected based on the product which has reached into clinical and then into the market. So this is how the framework, the ecosystem that we have developed is transferring us into the personalized four frameworks, and due to those personalized framework, we actually have moved into the uh, in a step where we can actually go and utilize it for the personalized. Now, what are our impacts have been created, and what is our vision utilizing this framework for the personalized medicine? So majorly it is the transforming of the global cancer prevention through the personalized herbal medicine. So there is already so many herbal medicines, but it is very difficult to say that which molecule is hitting which target and which can be utilized in the cancer prevention. That's where we actually utilizing this four framework and transferring this global cancer prevention strategy into that. So when we look at the traditional approach, um, the personalized formulation approach is actually shifting the paradigm into the cancer prevention, where the generic supplements are now going to be more precise, target-based natural compound. So in the traditional approach, where we are looking at one size fit supplement, and due to that, what is happening, there is a limitation of personalization, and also it is going to be difficult with the efficacy. Simultaneously, the safety is also sometime issue because it's not going to fit to all people. So with the Swalife innovation approach, we are utilizing AI-driven personalized formulation, which is going to validate the targets and going to optimize the bioavailability. As I gave you one of the example, means yes, nano is one of the technology, but simultaneously having different small molecules combinations is also decreasing some of the absorption or viability of the compound, and that we are doing with the AI innovation, which we think can be utilized for the validation and optimization of the targets and the lead. So the impact we want to build is that we want to have a global pipeline of the development of the precision herbal products, which can actually utilize in various cancer um, prevention strategies. So in the first phase, what we have focused on is that the breast cancer and oral cancer, where we are utilizing for preventive formulation. Now, the example which I gave you in vivo is about both the things. It's about preventive formulation, but simultaneously, we are also focusing on the chemo prevention of the side effect produced due to the chemotherapy drug. So it is like, yeah, it's not only that the herbal product is going to be given separately for prevention, but simultaneously when the cancer patient chemotherapy is going on, the herbal formulation can be given, so that way it can actually reduce that side effect as well. So that is where the foundation. The second, we are now moving into major other cancers as well. So there will be expansion into the other cancers like lung, prostate cancers, and other cancers. So that way we can actually develop the uh, product for that purpose, utilizing our AI platform and the four framework which we use for the personalized formulation. And then we will be focusing on the international market expansion and regulatory approach. So majorly at the moment, our focus Indian market and African market, but simultaneously we are into Europe and UK market. So we are focusing that international market as well. And utilizing our AI tools, we will be focusing on the innovation where we can actually have a precision herbal product which can be utilized in different cancer for the prevention globally. So that's where the global pipeline development is our major focus on. When we look at the market impact projection, as I already talked about the cancer burden, uh, when we look from the product point of view, the market opportunity, the size is actually about $50 billion, which is a cancer preventive market. That is about for 2030, and about 25% there is a prevention potential which can be utilized for reduction in the cancer in incidence through the prevention. So it is going to impact about 1 million plus life when you target the numbers of individual that you are going to serve in that 30 till 2030. So that is where the market is projecting the preventive market. We are looking different collaborations, majorly the clinical collaborations are there, like doctors and all the hospitals we are focusing on. But simultaneously, one of the collaboration I am continuously, so in 2017, when I came back to India, I am simultaneously focusing on many of the academic partners, even the universities and the research institute, where we can actually collaborate and utilize the platform and validate the clinical activity of this innovation. So majorly when we're looking at the academic partner, it is for the clinical validation purpose. So we will be setting the cancer centers at the at the academic level. In addition to that, as I told you, the doctors, the healthcare providers like hospitals, we are already having tie-up with them for our telemedicine and doctor insight division. We are also having a strategic partnership with different pharmaceutical and nutraceutical companies where we can actually have the minimum viable product for our personalized strategies, but simultaneously when we need the scaling up, we also having that partnership with the companies. So the regulatory strategies, as I already mentioned, that the Asian market and the African market, we are already in those markets. But in addition to that, now we are focusing on the US market entry through this dietary supplement regulation. The European market is actually very strict, and where the compliance is there is a lot of thing being needed for that compliance. So definitely we will be focusing on that. But international standardization we are utilizing, so that way we can actually focus on different markets in the world. When we look at the future innovation roadmap, majorly there are a lot of things happening in technology point of view. So AI is one thing, but again, AI is like a tool where uh, you need to understand that how it works. So based on that, the science which you are doing in wet lab can be utilized with the help of AI, and the knowledge can be grown and expanded. So technologies are developing and we need to adopt them. So one of the quantum computing, where the molecular modeling is going to be impact, is another area. Then pharmacogenomics, testing genetics of the person will going to give you the specific target of that person. So this is going to happen, and a lot of information can be utilized to develop the molecules and also the formulation. So pharmacogenomic is important. Then integrating biomarkers. So when we look at the biomarkers, the real-time health monitoring is important, which is going to help to adjust the formulation. So pharmacogenomics, biomarking integration is going to help to reduce the further disease complication, but simultaneously it can be utilized in the prevention as well. And also application beyond cancer to different chronic diseases, as I mentioned that in the prevention category and with the AI platform. Here I mention about small molecules, secondary metabolite from plant, but it can be utilized and for the cancer, but it can be utilized. The platform can be utilized for small molecules. It can be synthetic. It can be drug repurposing purpose also, and not only for cancer, it can be utilized for other chronic diseases as well. So there is that also need to be understand, because what is happening most of the time, the pharma companies are focusing on specific pathways that we like a target base for certain diseases, and then after like 4 5 years, it is understanding the target is not actually have been hit by the molecule. So all that studies, all that financial burden is coming on the pharma. But now with these strategies, with the startups, it can be actually collaborated. So molecule can be out licensed to other company, and different products, different targets of different disease can be hit by the other companies where the molecules has been out licensed. So these strategies can be done. So therapeutic expansion can be done. So where we are looking as a goal of Swalife Biotech is to be a global leader with preventive oncology utilizing the AI. So we are actually setting standards here for the personalized um, natural medicine. So starting with the scientific foundation and utilizing the technology innovation, we are moving into the market expansion and moving into the global leadership of AI-driven preventive oncology. So that's where we are looking ourselves. So that's from my side regarding the um, personalized medicine in herbal formulation, and how Swalife is actually transforming this cancer utilizing the personalized herbal science. So yes, together we can prevent cancer before it actually starts. That's what the aim is of Swalife Biotech. Thank you. If you have any question, I'm happy to answer. Over to you, Shri and
Thank you, sir.
Saj.
Actually, we have some questions from our viewers.
Mhm.
Like, first question is, are genomic or metabolic profiles used to guide herbal selection in personalized cancer prevention?
Sorry, can you repeat it again?
Yes.
Yeah.
The first question is, are genomic or metabolic profile
Genomic.
used to guide herbal prevention, selection?
Yeah, so as I mentioned in the the point of pharmacogenomics, yes, the genomics can actually can be utilized. It can be utilized in the selection of the plant or utilized for the plant extraction. So let's just give you an example. When we look at the network pharmacology, you can actually decide what type of extraction can be done uh, in the plant based on the targets which you got in the network pharmacology. So yes, the pharmacogenomics can be utilized in the selection of the plant, in the selection of the extraction, in the selection of the small molecules. It depends on what is your aim when you are actually focusing on utilizing the genomics.
Okay, thank you, sir. I hope the viewer got his answer. Uh, Shresh, do you have a next question?
Hello.
Yeah, yeah.
So there is a next question. 1 minute. The next question is, can you please explain how the Swalife AI tools like toxicological predictor, herb disease mapper, or the gene expression tool can be useful for students and researchers in a real project?
Okay, so so basically that's where our aim of having a collaboration comes. So as I told you that AI is a tool, it is not going to work on itself. So we need to utilize ourselves when you are actually using the AI tool. So I'm just giving you the background. So let's say, for example, if you look at the a common example of ChatGPT. When you ask a question to ChatGPT, it is going to give you answer. It doesn't matter what question you are asking. But if you are asking it a proper questions, it is going to give you a proper answer, which is going to link to different areas. So just recently, we actually developed a prompt studio. So this prompt studio purpose was for this, where let's say, for example, you are doing a drug discovery. In drug discovery, the major part is the target. The target, if you get the target wrong, it is very difficult that you will have a lead in the future, and that's what is happening. But then how to get the target? So there is data already available, but simultaneously the genomics where the gene sequencing can be done and we can identify the target. So to answer your question that how the Swalife AI tools. So in case of the toxicology prediction, it is important to understand that what the proteins or the targets are actually going to increase or decrease when this molecule is given to the human or to the animal. That information can be isolated with the AI, and for that purpose the AI is connected to the LLM. So there are LLMs like ChatGPT, Gemini, then Perplexity, and they are actually giving us a generative information. A generative information is not a wrong information. It is actually based on the information which is there in the public domain, and it is generating that. So it is, we cannot say that it is 100% right, but it is not 100% wrong as well. So those tools are going to help us to understand that how the toxicity can be predicted, how the herbal disease mapping, which we showed in the network pharmacology, that the molecules from the herbs are actually targeting the protein in different diseases. So we can actually map that. So if there is a molecule which is hitting one of the protein, showing a binding affinity high than the standard drug, we can utilize that. But again, when you are actually also using another molecule, where I gave you an example of curcumin and naringin. So if you add naringin, it is actually increasing the viability of the curcumin. So this, this information is all coming through this AI platform, this network, and this mapping is going to help to understand. Now, how this can be utilized for students or researcher is by, as I told you at the initial only, that Swalife is interested that when you want to increase the boundaries of science, you have to have a collaboration, and that collaboration can actually help the students and also the researcher to understand this tool. So we will be collaborating, and we can actually use those tools with the students and the researchers.
Okay, thank you, sir.
What else?
So there is one more question
Mhm.
from a viewer. In nanocore technology on for herbal medicine and other type of formulation, like what type of nanotechnology is used?
So majorly, what so like with our case, we actually, when we started with the small molecules from plant, we were not actually interested, or we were not giving a thought for nanotechnology. But what happened is that when you look at the small molecules from plant, molecules like non-polar, most of the time there is a issue of absorption, viability, and those type of molecules need to then actually move into the nanotechnology or nanotechnology type of the dosage form. So in this case, you can actually any molecule which you think that is playing important role, but due to the absorption or due to the viability, it is not showing its effect in the body. You can actually create nanoparticles. You can create nano suspensions are there, emulsions are there. There are many now nano different types of nano dosage forms can be created, which are useful to utilize those molecules which are not showing good activity when they are used individually. So even when we look at the curcumin, it can be put into nano, and it can be utilized to increase the viability.
Okay. Uh, Thank you very much, sir, for
Thank you.
an enriching and thoughtful session. I hope viewers got their answer for their questions. Your perspective on integrating herbal sciences with precision medicine was truly eye-opening.
Hello. Yes, sir. You talk has given us valuable insights into how ancient herbal knowledge can be personalized for modern day challenges like cancer. I'm sure our participants found it equally inspiring, and we guess about the questions and the answer segment also. And there is a various information we are gaining, like what are the gapping bridges, then what are the histological study, study design, and what are the behavior study, and what the use in the modern discovery, such as the like a cancer prediction or the treatment of a cancer. Thank you, sir.
Thank you. Thank you. Thank you, Sri and Sanjana and the organizers.
Before starting the next session, that is for of poster presentation session, I would like to rememorize the rules. That like there will be two rules mainly. 10 minutes will be given for the representation time of the poster, and 5 minutes for the question answer round. So in total, there will be 15 minutes for every participate for representing their posters. So, starting with the poster presentation. Good morning, everyone. It's my pleasure to welcome you all in the poster presentation session. This is the platform where research ideas take visual form and inspire us with new direction and pharmaceutical sciences. In this session, we will hear from our first excellent presentator, each contributing unique insight through their research work. Let's begin with Patel Priyaben Ashok Kumar, our first presenter, a postgraduate student from Parol Institute of Pharmacy and Research, Gujarat. Her research poster is titled with From Traditional OTC Herbs to Modern Antimicrobial Hits, Atomic Validation of Phytochemicals Targeting DNA Gyrase and Topoisomerase IV. Her research poster, under the guidance of Mr. Sachin Kumar Sharma and Dr. Pinkal Patel, her work bridges traditional herbal wisdom with advanced computational validation, aiming at novel antimicrobial target. Let's welcome Priya Ben as she presents her innovative study.
Good morning, ma'am.
Good morning.
Can I share my screen?
Yes, you can.
Screen is visible, ma'am.
No, the screen is. Yes, it is visible now.
Okay. Hello, everyone. My name is Priya Patel, and I'm excited to present our work titled From and Traditional OTC Herbs to Modern Antimicrobial Hits, Atomic Validation of Phytochemicals Targeting DNA Gyrase and Topoisomerase 4. Before starting the presentation, I would like to start my presentation with a Sanskrit slok called as Yat Kinchit Aushadam Loke Sarvam Ashwagandhikam. Whatever the medicine exists in the world, Ashwagandha is part of it. Today, we face a significant challenge is the rise of antimicrobial resistance. As bacteria evolve, our current antibiotics are becoming very less effective. Our goal was to find a new way to fight these superbugs and we turned into ancient source, and the plant is ashwagandha. We also know it as Virania somnifera, and this plant has been widely used for centuries in Ayurvedic medicine. We wanted to scientifically validate its potential using modern computational drug discovery. We focused on two bacterial enzymes, DNA gyrase and topoisomerase 4. As we all know, DNA gyrase and topoisomerase 4 both are the best antibacterial targets, and that's why I am focusing on both the targets. Our approach was systematic and comprehensive. We curated a data set of 223 223 phytoconstituents from Bidania somnifera to check its antimicrobial potential against DNA gyrase and topoisomerase 4. And for this, we have used main and molecular docking technique, and along with it, we have performed its ADMET screening with a Swiss ADME database, and for that, we have prepared a database of 223 phytoconstituent, and it will bind to our target proteins, DNA gyrase and topoisomerase 4. The computational method allows us to predict the strength of the interaction or binding affinity before ever stepping into a wet lab. And we didn't stop there. We also performed ADMET screening to predict the compound's pharmacokinetic properties such as its absorption, distribution, metabolism, excretion, and toxicity. This is crucial step to ensure that any potential drug candidates are not only effective and not only binding, but it is also safe for using. And let's go for methodology. For methodology, first of all, we have prepared protein with the use of RTSB PDB. And after selection of protein, we have validated this protein with PDB sum software. And by getting the Ramachandran plot of these two different targets PDB, and these both are the Ramachandran plots of these both proteins. First one is the DNA gyrase, and the second one is for topoisomerase 4. Data set preparation, we have taken data sets from different database like as PubChem, Impact, and Dr. Duke database. And from that, we have taken 223 phytoconstituents of Vidania somnifera. And with these 223 phytoconstituents, we have performed it's a molecular docking and Swiss ADME characteristics. Molecular docking was performed with order of Vina software, and with Discovery Studio, we have we get amino acid interaction images. It is the overall workflow of our work. First one is herbal source selection. In this, we have selected 223 compound data set. After that, ligand preparation, ligand was prepared in Chem3D software, and after that, we have minimized its energy in same software. And along with this protein selection and preparation, protein was selected from RCSB PDB website. And for DNA gyrase B, I have selected 5 mmn PDB ID, and for topoisomerase 4, 3K9F PDB ID was selected. After that, we have go from molecular docking conducted using Autodoc Vena software. And after that, amino acid interaction visualization was done by Discovery Studio. And then we have performed ADMET prediction of compound. By doing the 223 compounds molecular docking study, the best docking score compounds results are shown here with 3K9MS and 5 mmn. Vidanolite T. Vidanolite E shows a better binding affinity as compared to other constituents. Along with it is also compared with ciprofloxacin and novobiocin. Because of this, both are validated drugs which are used in market currently. And vidanolite E shows better binding affinity in comparison with this both marketed drugs also. Along with this, the amino acid interaction images are also prepared with the same PDB ID, and it is also performed for vidanolite E because it is the potent binding, it shows the potent binding affinity with DNA gyrase and topoisomerase 4, and its amino acid interactions are also compared with ciprofloxacin. And it is the MDS data. MDS data was performed for stability testing of this particular compounds, and for this low RMSD and RMSS values are observed. These values indicate that the protein ligand complex remains stable throughout the simulation, supporting the strong binding affinity we observed in our docking studies. And it is the ADMET screening data set, and it is also for this constituent which composite docking studies are shown because of other constituent ADMET screening are also performed. And from boil image, we can see that all the constituents are unable to cross the blood-brain barrier, and it is the Lipinski rule of 5 data of these particular compounds. And in this, vidanolite C is the only compound which show Lipinski rule violation. Our research provides scientific basis for traditional use of ashwagandha and demonstrate how modern computational method can accelerate the discovery of new drugs. The next logical step is to perform in vitro and in vivo experiment to validate this finding and move closer to developing the novel antibiotic. Thank you so much. I am happy to take any questions.
Hello.
Hello.
Yeah, it was nice presentation, Priya. Can you tell me what in vivo assay you are planning to do in future, or where you see this project after 3 years?
After 3 years, after this, I want to perform. Firstly, I want to perform its network pharmacology also, because of after network pharmacology, I will confirm it was actually showing the network on DNA gyrase and topoisomerase 4 or not. After it, I will go for its these compounds extraction and all the methodology. And if it is possible for me, I will go for its formulation. And also, if it is able to show and if it is possible to perform in our lab, I will definitely go for it.
So on what basis you selected the protein?
On basis of firstly, I have selected the targets, the DNA gyrase and topoisomerase 4, because it is a better antibacterial target. After that, I have for selection in RCSB PDB, I have checked its resolution. After resolution, I have checked its species, on which species it is available in website. And after that, also validated through Ramachandran plot also, because the Ramachandran plot will show accurate validation of the protein. And after a validation, I have also confirmed docking proteins is able proteins confirmed by docking with ciprofloxacin and novobiocin.
Okay, in vivo assays?
In vivo assay is not done.
Yeah, but are you
Actually,
planning?
I am currently focusing on in silico. Currently, I am focusing on in silico. If it is possible in future, I will be planned for in vivo.
Okay, thank you. Thank you.
Thank you, sir.
Uh, Priya Ben, we have some question from a viewer. Like, what are phytochemicals being invested as potential antimicrobial agents?
In ashwagandha, I have in ashwagandha for the total 223 phytoconstituent, vidanolide E is the most potent. By in silico studies, I have confirmed it.
Okay.
Along with it, there are so many phytoconstituents, as we all know, neem, curcumin, and so many plants are available to show its antimicrobial affinity. But in this particular, I have selected DNA gyrase and topoisomerase for both because of it. I want to target dual. I want to go for dual targeting. Along with this, there is so many work reported on DNA gyrase, but there is no work reported on topoisomerase 4 with ashwagandha. That's why I have gone with, I have worked with both target and I am targeting with both. And it will be confirmed by network pharmacology by in future.
Okay, and how were the active phytoconstituents selected for screening?
I have selected I have selected total 223 phytoconstituents from different databases, like I have mentioned already, PubChem, Dr. Duke, and Impact. From with this 223 phytoconstituent, I have performed the molecular docking study, and with the best molecular docking score molecular docking score, I have taken some of here. From all compounds docking, I have selected here. And from this, I have also performed the ADMET screening of this compound, and ADMET also shown the best docking score is done. But from best docking score, also vidanolide C is unable to cross the Lipinski rule. Lipinski rule violation is able to see in vidanolide C.
And which class of compounds was found to be most active?
Sorry, ma'am.
Which class of compounds was found to be most active, like alkaloids, flavonoids, or which one?
Mostly, as I have a review, I have observed in review. Mostly, I have got literature on mainly on flavonoids. I have observed more articles.
Okay. And one last question. How does atomic validation confirms the reliability of docking result for phytochemicals binding to DNA gyrase?
Ma'am, please repeat.
How does atomic validation confirms the reliability of docking result for phytochemicals binding to DNA gyrase?
It will not confirm. It will give results and readings idea upon according to it. If atomic validation means I have validated atoms and also it, and from this molecular docking is not only source, and it is not like I have performed molecular docking and I will confirm it is better. For that, I will go for ADMET. Also, I have to go for MDS screening. Along with it, I have to perform its toxicity data and also go for bioactivity screening, 3D QSAR. There are so many methods, and after performing all these in silico methodology, I will confirm this particular constituent is best because it shows best activity in all these methods. I will perform all the methodology, not only this docking, but I will go for ADME and 3D QSAR. After that, I will go for MDS screening and network pharmacology. After doing all these things, I will confirm this particular constituent is better. And from selection of 5 to 7 these constituents, I will go for its in vitro and in vivo studies. And after that, I will confirm, yes, it is the best compound, not from docking only.
Okay. Thank you, Priyaben, for the enlightening presentation. Your research beautifully connects traditional knowledge with modern molecular sciences. Thank you very much.
Thank you so much.
Yeah. For the next poster presentation, I would like to call Dr. Bhavna Utkarsha Jain. She brings her valuable expertise and strong academic research experience. Her poster reflects her deep understanding of the pharmaceutical fields and highlights key aspects of her ongoing work. Let us know and listen to Dr. Bhavna Jain as she shares her finding. Dr. Bhavna Jain.
Hello, am I audible, ma'am?
Yes, ma'am, you are audible.
Hello. Screen is visible to all? Hello.
No, ma'am, your screen is not visible yet.
Is it visible? Hello. Hello.
Yes, ma'am, the screen is visible now.
Hello.
Hello. Just wait for 2 minutes. Share my screen soon. Is it visible?
Yes. Just maximize that screen.
Hello.
Ma'am,
Just
can you please open the presentation mode? Like the screen is visible very like in smaller one.
Yes. Myself, Bhavna Uttad Jain. Today I am deliver a poster presentation on the topic of molecular docking, cytotoxicity and antioxidant evaluation on bioactive compound from Caparis zalanica lean stem. I'm using here lean stem of plant, Caparis zalanica plant and stem part of that particular plant. And introduction, in case of introduction point of view, herbal plant, these are the most important aspects related to the Indian peoples, and most of the Indian peoples, they are using herbal plant for the curing the lots of drugs, and that's why I'm using a Caparis zalanica. Here in Marathi, it's called as a Vaghati and belong in the family Caparisis, and plant shows a number of phytoconstituents just like alkaloid, flavonoid, and phenolic compound. And pharmacological activity point of view, it shows anti-cancer, anti-cancer as well as antioxidants activity because it contains alkaloid as well as phenolic compound. Flavonoid is also related to the anti-cancer activity. And today my topic aim is to access the phytochemical content and investigate the therapeutic potential of Caparis thalanica stem and its pharmacological benefits. Object to prepare the on extract plant stem, isolate and characterization of identify the phytoconstituent to evaluate the pharmacological activity related to anti-cancer as well as antioxidant activity, analyze binding affinity related to isolate through on in silico docking study. I am using here the SMINA software for docking study, and in case of experimental work, first of all, plant is authenticated by Botanical Survey of India in Pune, Maharashtra, and extraction with the help of pet ether, chloroform, and ethanol. These are the successive extraction. After that, chromatographic analysis with the help of TLC and HPTLC and column chromatography. And next, pharmacological screening with the help of anti-cancer activity as well as antioxidant activity. Next, in case of result and discussion, first of all, ethanolic extract first use for the next activity point of view, because of chloroform and pet ether not useful because it shows less phytoconstituent as well as pharmacological phytoconstituent. And here for the TLC point of view, silica gel used as a stationary phase, mobile phase, and hexane and ethyl acetate 8:2. For the calculation point of view, retention factors, distance solid by solvent divided distance traveled by solid divided by solvent in centimeter, and these are the chromatographic study. First of all, TLC, I found only 3 spots, and next HPTLC done at 263 nanometer. According to the HPTLC, I found that it may contain alkaloid, and after the analysis, after the isolation study, I'm choosing here the column chromatography. And after the column chromatography, different fractions are mainly used for the analysis point of view. That is the IR, NMR, and mass. Next, for with the help of IR, NMR mass study, I found this structure, isolated compound structure, and that structure also correlate with brucine. And after that, to perform 3D structure of extracted compound as well as standard. I'm using here the standard compound, that is osimertinib, and here the ligand used 2JIU. These are the ligands related to the epidermal growth factor receptor. These are related to the non-small lung cancer cell, and these are the non-small lung cancer cell means those patients who are not smoking at that point. That is the EGFR mainly used. And these are the RMSD study as well as the RMSF study. In case of RMSD study, root means standard deviation study. In this case, that is the carbon compound means blue lines are there, and green pink lines are the beta helical compound. These are whatever the standard means lines means. These are related to the whatever the compounds they are fluctuated or not. These are the means carbon helical compounds. These are the beta lines, the mostly rigid one. That's why it confirmed that whatever the compound, their stability is more. I'm choosing here the 3 cell line, that is a wild type EGFR. Second, that is a wild type threonine, that is a 790 to methionine at complex, and last one, that is a these are the complex. It's a mutant complex of these 3 cell lines, and in this case, that is the first compound, that is a wild type EGFR means HCC 820 cell line. It shows more potent in case of RMSD study as well as in case of fluctuation RM root means standard fluctuation study. In this fluctuation study, I'm again choosing the 3 cell line, that is the extracted compound of, that is a wild type EGFR complex. After that, extract a compound of, that is a EGFR, that is a threonine to methionine at the 790 position complex, and third one, that is a mutation complex. And in this case, that is the first one, that is the HCC 827. That means wild type EGFR shows a better rigidity as well as it shows a 100 nanosecond. These are the green line indicate its protein binding, and red pink line it shows the whatever the compound. These are the more stability in case of first one than that of the other. That's why HCC 827 as well as wild type EGFR shows a more potency than that of the other. Next, according to that, that is the pharmacological activity with the help of MTT assay study. MTT means related to the formazole. Formazole, it shows a purple color, and it is in case of chemical point of view 3,4,5 trimethyl thiodiazole 2YL formazole. In this case, that is I'm choosing that is a 3 cell line, and it shows a better these graphs related to the percentage cell inhibitions related to the concentrations, and in this graph shows the HCC 827 shows a more potency than that of the other. Next, anti-cancer related to the same antioxidant activities, same uh, same same cell line as well as extracted as and isolated compound shows to perform the activity. In this case, the first one, that is the isolated compound, shows more potency than that of the standard one. Here I'm using here the standard as a ascorbic acid. After that, Western blot assay, but Western blot assay related to the which type of proteins or which compound is more potency related to the particular receptor, and here the related to the EGFR, phosphorylated EGFR, and phosphorylated AKT kinase, and beta actin as a control. In this case, phosphorylated and phosphorylated EGFR as well as phosphorylated AKT. It shows the best signaling pathway. Means these are the I'm choosing here the HCC 827 means wild type EGFR, and at that point that is a both the type, that is the phosphorylated EGFR and phosphorylated AKT shows a positive response related to the downstream signaling pathway, means decreasing cancerous cells. That's why Western blot again confirmed that HCC 827 means wild type EGFR shows
You have
a
only 1
potency.
minute left.
Hello.
You have only 1 minute left. Now
Next,
continue.
Ethanolic extract of Caprizalanica stem is a rich source of biotic compounds including alkaloid, flavonoid, and phenolic compound. Next, anti-cancer activity was assessed, that is HCC 827 H1 975 means threonine to the methionine at 795, and HT29, that is the third cell line, means this is a combination of these three. These are the mainly 3 cell lines. This is the cell lines showing strong cytotoxicity, means anti-cancer activity, especially against H29 ethanolic extract of Caprizalanica effectively, and autophosphorylation with the comparable efficiency with osimertinib as a standard compound related to the anti-cancer, suggesting modulation of EGFR and AKT signaling pathway. Molecular docking studies confirm strong binding and wild type EGFR mutant EGFR forms, and HCC 827 confirmed the protein help the Western blot assay, showing IC50 values comparable with the standard ascorbic acid. In case of future prospect point of view, this may be related to the means particles related to you have to form any lung cancer therapy, as well as this drug may be used in case of inhalation point of view for the cancer therapy, because these drugs is related to the EGFR, means epidermal growth factors that are related to the lung cancer. Those patients who are not chain smokers, at that point this mainly symptoms are followed. Presentation is over. These are some references I have referred for this presentation. Hello. Hello. Am I audible?
Hello. Yeah. Thank you. It was nice presentation, Dr. Jain. Just a question on the cell line. Based on what you decided to select these 3 cell lines?
What? Pardon, sir. What you?
For the MTT assay, why did you selected this 3 cell line?
Because all means most of the cell lines are used for the MTTSA, because MTTSA is a well-known anti-cancer activity in vitro. That's why I'm choosing a different cell line, EGFR, epidermal growth factor receptors. They are related to the non-small lung cancer cells. That's why I'm choosing different cell lines than that of the other.
Now, why didn't you use HEBG2 cell line for MTTSA?
Why you?
Why didn't you use HEBG2 cell line? Liver cancer cell line.
liver cancer cell line. No, sir. These are the non-small lung cancer cell lines. These are the related to the lung cancer.
Yeah, but you are not planning to, the topic is not relevant to lung cancer, right?
EGF means wild type EGFR means that cell lines related to the lung cancer. Cytotoxicity is related to cancer. That's why means cytotoxicity related. You can do it any type of cytotoxicity means any organs means whatever the intestine cancer, lung cancer or skin cancer. That's why I'm choosing this lung cancer.
Okay, so in your future prospect, you mentioned that exploring the potential of zinc oxide nanoparticle.
Yes. Yes.
Do you done a literature where zinc oxide
Yes.
particle,
Yes.
nanoparticle?
I found that one paper related to this zinc oxide nanoparticles, but and same work on means Caparizallanica. They are using leaves extractions. I'm using here the stem part of the plants. I found that paper in
Yeah.
2000.
But what I'm trying to say here is zinc oxide
Possible, sir.
and titanium oxides are used in sunscreen as
Yes.
a nanoparticles, and they are actually creating DNA damage due to which cancer is occurring. So you need to think about the zinc oxide nanoparticles. Thank you.
Thank you so much, sir.
Uh, Hello, ma'am. Actually, there is one more question from our YouTube audience. How can antioxidant or phytochemicals contribute to anti-cancer or antimicrobial properties? Can I repeat the question? My question is, how can antioxidant phytochemicals contribute to anti-cancer or antimicrobial?
Flavonoid, alkaloids are the phytoconstituent mainly responsible for. These are the some phytoconstituents that are present already in that particular plant. Hello.
Okay, thank you, ma'am. Thank you, Bhavna Jain, ma'am, for your thoughtful and impactful presentation.
Thank you so much.
Uh, our next presenter in this session is Dr. Dishraj Shamkant Tsumble from Amrutvani College of Pharmacy, Sangamner, Maharashtra. His poster title is Evaluation of Cassia Orticella lean flower for its diuretic efficacy. This important study, guided by Dr. Manchandra J. Chavan, focuses on validating the traditional medicine use of Cassia or Orlicata in managing diuretic activity. Let's welcome Dr. Tsumble to present his significant contribution. Dr. Dishraj. Uh, Dr. Tsumble, is he connected?
I don't think he's there in the participants.
Uh,
So we will just wait a couple of minutes more.
okay, so can we take the next candidate, or we should wait, sir?
We will wait. We will wait.
Okay, sir.
Sanjana, if the next candidate is there, we can continue then.
Uh, okay, sir. Shreyash, can you please take it on?
Yes, thank you, Sanjana. The fourth candidate was is Shraddha Vilas Patil. Is she present?
Yes,
Shraddha?
I'm present. Yes, I'm
Okay.
present.
Okay, thank you, Sanjana. Now, I would like to introduce our next poster presentation. That is Miss Shraddha Vilas Patil, a postgraduate student from the Department of Quality Assurance, Bharati Vidyapith College of Pharmacy, Navi Mumbai. She will present her research in SPF-TLC fingerprinting and evolution of wound healing potential of lotus leaf extract loaded nanofiber patches. Her work carries out under the guidance of Dr. Sneha Agrawal, combines advanced nanofiber technology with herbal extract to explore new wound healing therapies. Let us welcome Ms. Shraddha Vilas Patil. Shraddha, you can now start your presentation. Hello. You have 10 minutes for presentations and 5 minutes for the question answer session.
Am I audible?
Yes, you are audible. Start.
Am I screen is visible?
Yes, screen is also visible.
Good morning, everyone. Myself Shraddha Patil from Bharati Vidyapith College of Pharmacy. So today I will presenting on the HPTLC fingerprinting and evaluation of wound healing potential of lotus leaf extract loaded in nanofiber patches. Basically, the wound healing is a complex biological process. Delay wound healing is a significant clinical changes like diabetes or infections. The herbal medicine is contain the more amount of bioactive compound with the antioxidant and antimicrobial properties, which can promote the natural healing with fewer side effects. So we focus on lotus leaf extract because of there is a traditional medicinal use and high antioxidant content. So we developed the nanofiber packs containing lotus leaf extract and evaluating the wound healing. So basically, introduction, the wound healing is overlapping with 4 stages. First, hemostasis, which clots stop bleeding. Then proliferation, to immune immune cells clear the debris and fight infections. And in inflammation and proliferation, the new tissue is generated like granulation tissue, which form and to close the wound. And in remolding, we strengthen the newly formed tissue. So disturbing this stage due to the oxidative stress or infection can result in chronic wound.
Hello.
Sorry. Hello.
Good morning, madam. Myself, Dr. Deshraj.
Hello,
Wait.
sir. Just one is going on. After this, we will continue with yours.
Okay. So, okay. Okay, sir. Okay, no problem.
So shall we continue?
Yes. Continue.
So the herbal plants are supposed to healing by the reducing inflammation and promoting the fibroblast growth and enhancing collagen production, and it also acts as an antimicrobial. The lotus leaves are particularly rich in flavonoids, phenolic compound, and antioxidants, which promote the strong antioxidant and regenerative effect. So moving forward to the methodology. So first we perform the extraction. So the fresh leaf is collected and it's dry, and they converted into the coarse powder. This coarse powder is extracted with ethanol using subgillate apparatus to isolate the bioactive phytochemicals, and to confirm this presence of bioactive compounds, we performing the HPLC fingerprinting using the mobile phase, chloroform, ethyl acetate, methanol, and glacial acetic acid at ratio 6.5, 1, 2.5, 0.025 volume by volume. So at 366, we detected the 7 compounds with RS value 0.001 19 to 0.84, and at 254 nanometer, we detected 10 compounds with RS value 0.20 to 0.87. Then we prepared the nanofiber patch preparation. The extract was incorporated into nanofiber patches using electrospinning. So the polymer solution is prepared by a mixture of PVP K19 and ethyl cellulose is dissolved in ethanol, and the lotus leaf extract is gradually or slowly added into this mixture using the electrospinning at 50 to 20 voltage. We produce the thin uniform nanofiber patch suitable for topical application. Then further we 3 into 3 centimeter patch we made for the treatment and the placebo group. Now moving forward, evaluation parameters. By the DOE experiment, we selected the 4 formulation. From that, we selected F3 formulation because it gives a smooth and uniform thin patch, and their average thickness is 0.125, and the folding impedance of each batch is about 300. So it's indicating good flexibility and stability. And the drug was incorporated 5 ml of ethanolic extract, and their dose is 100 mg per ml was incorporated from nanofiber patch. Then moving forward to result, the wood healing activity, the incision model used for mixture rat, and then the rat is divided into the four groups: control, direct extract, ethanolic extract loaded nanofiber patches, placebo nanofiber patches. So first we observe the visual observation. In this, the wood healing was visually accessed by day 1, 4, 8, and 12. The ethanolic extract loaded nanofiber patches show the fastest healing with significant contraction by the day 4 and nearly complete closure by the day 12. And the direct extract group is also shows some healing when placebo and control group show some slower recovery. And in the percentage wood healing closure, the percentage wood healing closure, ethanolic extract of nanofiber patches show the higher percentage wood healing at by day 12, 99%, day 8, 75%, and day 4, 30%. And also in direct extract, they significant show some progress healing in day 12, 85%, day 8, 65%, and day 4, 25%. But in control and placebo group, show some slower percent wood closure. And the day of epithelialization, the ethanolic extract loaded nanofiber patch, the complete epithelialization showing day 9, and in other group show control in day 16, direct extract in day 11, and placebo patches in day 14. So it enhances the bioactive release and promoting the cell proliferation and tissue regeneration. So that result is indicate that the nanofiber patch loaded with ethanolic extract of lotus leaf is associated with the wound contraction and epithelialization compared to the other groups. The conclusion, the ethanolic extract of lotus leaf is by enhance the wound contraction and epithelialization in both direct extract and nanofiber patch groups. The bioactive compound is also accelerated tissue generation through the antioxidant and regenerative mechanism. The incorporation of the nanofiber-based formulation is further improved the efficacy by enabling sustained release and resulting in the superior yield outcome. So results indicate that the lotus leaf ethanol extract is a promising candidate for the wood management and highlight the potential of herbal nanofiber formulation. This was the references was the refer for our study. Thank you so much for your time and attention.
Thank you.
Hello.
Thank you, Shraddha.
Thank you.
So there are a couple of questions. One, why did you decided to use ethanol for extraction?
Because the ethanol is a good solvent and wide range of phytochemicals which extracted with ethanol, and it is a safe and non-toxic. So we use the ethanol.
So it is nothing to do with the focus of the molecules. So you are not focusing on polar compound.
Yes, we focusing also on polar compounds.
Okay, and what was the purpose of doing HPTLC fingerprinting?
Because then in their UV light, the bands they represent some different phytochemicals which present in the lotus leaf extract, and they're confirming that they're in the lotus leaf extract contain the major amount of bioactive compounds, and then the 10 compounds were detected. So the major bioactive compounds is alkaloids and the flavonoids, which give the antioxidant and anti-inflammatory properties. So we also HPTLC fingerprinting method is performed.
Okay, and in the wound healing assessment, there was no standard taken in the protocol.
Sir, which food drug is taken?
Which crude drug?
Family phenolic extract was lotus leaf.
No, but you are comparing, right? Wound healing. So there are drugs already in the market which are used for wound healing.
No, sir, I'm just doing the animal study on this food drug and check the wound healing activity.
Okay. And why did you decided to prepare nanofibers?
So because the nanofibers it gives the sustained drug release, it's also the moist healing environment, and it protects from the microbial contamination, and it enhances the drug penetration due to the large surface area.
Okay. Thank you. Thank you, Shraddha.
Thank you,
Shraddha,
sir.
hello.
Yes, sir. Yes, sir.
Thank you. There are some questions from the YouTube audience and the participants.
Yes, sir.
The first question is how does the HPTLC fingerprinting of lotus leaf extract help in standardizing the nanofiber patch formulation, and which major phytoconstituents were identified as a potential contributors to wound healing?
The lotus leaf extract, the alkaloid first primary contain the luciferin and its derivative non-luciferin, and the flavonoids contain quercetin and lutein is present. But in the market, there is a nanofibers are not available. So we perform the HPTLC fingerprinting.
Okay, and another question is what challenges exist in translating lotus extract loaded nanofiber wound healing dressing from laboratory research to clinical application? May I repeat the question?
Yes, sir.
What challenges exist in translating lotus extract loaded nanofiber wound dressing from laboratory research to clinical application?
Mhm. Application.
That means how?
It
What
requires
challenges?
the backing membrane required as a backing membrane, Hello, am I audible?
Yes, 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 yes.
The application of wood is it's required as a backing membrane and folding endurance
Okay,
to stick on the wood.
okay, okay. Thank you, Shraddha, for such detailed and innovative presentation. Your work on herbal nanofiber patches truly reflects the integration of traditional knowledge with modern pharmaceutical technology. Thank you.
Thank you, sir.
Our next presenter for this session is Dr. Dishraj Zumble from the Amrut Vani College of Pharmacy, Sangamner, Maharashtra. His poster title is Evaluation of Cassia auriculata lean flowers for its
Cassia
diuretic.
auriculata.
Yeah, sorry. This important study guided by the Dr. Manchandra J. Chavan focuses on validating the traditional medicinal use of cassia in managing diuretic activity.
Yes. Good morning to all of you. That hello, madam.
Yes, sir.
Presentation. I can share a presentation or
Yes,
what?
sir. You can share your screen.
Share. Okay. Uh, have you visible my poster?
Yes, sir. It is visible.
Okay. Okay. So can I start? Madam?
Yes, sir. Please.
So good afternoon, all of you. So myself, Dr. Deshraj, I am presenting my topic, that is the evaluation of Cassia auriculata lean flowers for its diuretic efficacy. So this specially ethnomedicinal herbs, that is the Cassia auriculata, belongs to the family Cisalpinaceae, and this family, it is a Cassia genus, locally or vernacularly known as in Marathi, that is a tarwad. So this flowers is a yellow color flower, and it is a ethnomedicinally traditionally useful for the specific for the kidney functions, urinary discharges, as well as diabetic patients, and also throat irritations. For that, this plants is a very useful one. This plant have taken for this present study specifically for their ethnomedicinal properties. So firstly, I have collected these flower plants, flowers of from these plants, and that extracted out with the help using the highly polar solvent, that is the ethanol, as well as water, distilled water, 70-30 ratio, and diuretic efficacy have evaluated on the specifically Vistar rats by using the furosemide. It is a standard drug for comparison have taken. This diuretic activity has determined by measuring the parameters such as urine volume, electrolyte concentration, pH of the urine. Such type of the parameters evaluated for checking the significance or screening of the hydroalcoholic extract of this Cassia auriculata plants. So this plant anciently, these herbs, it is having traditional property as a diuresis activity, and this traditional property, ethnomedicinal property related to the kidney issues also, and that people's patients suffering from the kidney issue, high blood pressure because of the that is a urinations, that is a lack of the urinations, and that is a reuptake of the number of, that is, electrolytes. So avoid that. These plants, or we can say as significance of this Cassia auriculata flower, useful for in this type of the complications such as urinary discharges, diabetics, as well as other also, that is a useful one. So these plants have taken, and it is already reported that these plants, it is having some more of phytochemicals such as flavonoids, phenolic compounds, as well as tannins, terpenoids, alkaloids, carbohydrates, and steroids. So my focus to explore these plants for these particular ethno-medicinal uses. So firstly, prepare the extract at 70-30 ratio into the ethanol and water, and that extracts have completely prepared the dried, then prepared the suspension into the again distilled water and check for their toxicity property. So acute toxicity studies, according to the OECD guidelines 423, I have performed, and then it was observed that this is a very efficacious. It is a does not having show no any toxic effect, adverse effect, no any mortality observed into the visitor rats. Then that number of group of they have taken, that is one group having six group of animals, and in that there are the four dividation of the groups. Group first has a control group, only giving the normal saline. Group 2 have taken for the standard drug furosemide as a diuretic drugs 10 milligram per kg per oral. Then group third taken for the extracts, and that extract of 250 milligram, and second dose that is a extract of the 500 milligram, such type of the group dividations into the animals. And then the dose have performed given to that animals, and after the administrations the animals were placed into the metabolic cages. So especially separate that is urine collections after the end of the that is a completion of this protocols, and that urine collected, and that urines it is a checked for their pH as well as electrolytes like as a sodium, potassium, and chlorine. Such type of the parameters I have performed. So in the that is a results have observed that the like as a when compared with the extract group with the control group as well as standard groups, so it was observed that so there are the increase the urinations as well as increase the excretion of like as a sodium, potassium, and chlorine in these samples. Then alkaline pH is also increased into the sample of the urine after the administration of the Cassia auriculata flower extracts. So this conclude that this having showing effect as per the traditional ethno-medicinal properties. And this hydroalcoholic extracts of when the 500 milligram per kg when we observed, so there is increase the urine volume 7.52 rather when the it is a compared with the control that is a 4.32. Similarly, sodium also potassium and chlorine in that also there are increased the volume of that excretions of that particular sodium, potassium, and chlorine volume in these samples. And pH is also have become alkaline, that is 8.84 into the hydroalcoholic extract of 500 milligram per kg when compared with the control groups. So this study have results have depicted that this Cassia auriculata flower having strong diuresis activity, and it is a very important beneficial for the such type of the patients belongs to the related to the high blood pressures and number of complications. So this concluding that this drug having it is a non-toxic because it is a herbal and it is a safer, as well as when we go through the evaluation of the acute toxicity study, it's showing that it is a safer, no any mortality is observed. Also, this drug it is a preferable rather than the synthetic drugs because synthetic drug showing maybe another complications adverse effect, but this herbal extract it is a very useful for significant diuretic activity. It is a safer, non-toxic, and also support concluding support that this traditionally ethno-medicinally this plant it is a diuresis, and it is a proved that evidently it is useful as a diuretic activity. I have go through the number of references also, and on that references I have found that this plant having ethno-medicinally important for these properties. And then later what I have taken for this study, and after the completion of the study I have proved I have come to the conclusion, yes, this plant is a it is a Cassia auriculata plant. It is a having diuresis activity. This study have performed in our animal house, in our college animal house, and in that I have uh, taken this facility from our college as well as support of our Sausta. I grateful that. Thank you, madam.
Thank you, sir. It was nice presentation. Just couple of questions.
Yes, yes.
So you created ethanol water extraction, right?
Correct,
Uh,
correct.
so what was the final quantity of the extract?
I have taken this it's a 70-30 ratio. In that is ethanol and water because of high polarity. And that I have taken fresh flower portions, and that is macerated. And after the macerations, I have filtered that extract and collected. So it is a properly up to that is a 20% of that extract have come taken, means in that proportions. So total power prepared materials I have taken that up.
And how was the extract concentrated? So did you do?
Yes, yes, maceration. Because of that maceration process, I have using the methodology by same, that is a cold way, that is a desiccator I have taken. And in the desiccator I put it, that is absorbers. And in that petri dish samples, I have put it this filtered portions of this extract. So easily that is after the 24 hours, some of the portions have reduced, and later word again it is reduced. So number of desiccator using, I have concentrated that samples, and it is a very so avoid the heating for denaturation of the compounds.
And was the did you use
Yes.
freeze dryer
Uh,
to
no.
remove the solvent?
After remove the solvent, it is a completely dried as such into the desiccators, and then later what that powder directly I have again put into the water for preparing the suspension.
Okay, with the animal protocol,
Yes, yes.
what was the the time frame for the protocol to finish?
Firstly, I have taken, so already we have the animal house. So in that we have, so in that protocol, no any sacrification. So that animals, firstly, I have isolated from the regular animals, and it is a fasted. So after the fasting, then later word, it is a that animals have taken the groups, and after the 24 hours fasting, then later word, that animals have suitably, it is a separate out into the separate cages for the according to the grouping treatments. And then later word, it is a after the treatment with our extracts as well as standard drug, then collected through the funnels. I have collected the urine for that evaluation next.
Okay, thank you, sir.
Okay, thank you, sir, for giving me this opportunity to presenting in this international conference. I very much thankful.
Uh, Thank you, sir. It was truly reinforcing the bridge between ethnopharmacology and evidence-based medicine. Further session will be continued by Shri Kadam.
Okay, thank you, Sanjana. We take the fourth participant, that is Shraddha Vilas Patil now. Moving forward, the next participant, Pranjali Navnasuta. Pranjali, hello.
Yes, sir.
Okay, our next presenter is the Miss Pranjali Navnasuta, also postgraduate student from the Department of Quality Assurance, Bharati Vidyapit, College of Pharmacy, Navi Mumbai. She will be presenting her project titled Formulation and the Evolution of Limonene-Loaded Solid Lipid Nanoparticles Against Candida albicans. This research, under the guidance of Dr. Sneha Agarwal, investigates how natural compounds like limonene, when formulated into lipid nanoparticles, can serve as effective antifungal agents. Let us welcome Ms. Pranjali Navnasuta.
Yes. Thank you, sir. Is my screen visible?
Yes, yes. Screen is visible now.
Okay. Good afternoon, everyone. So, my topic for poster presentation is formulation and evaluation of limonene-loaded solid lipid nanoparticles against Candida albicans.
So limonin is a phytoconstituent that belongs to terpene group. It is basically a monoterpene, and it is present in citrus essential oils. It has shown excellent antifungal as well as antibacterial activity. Here we used Candida albicans as it causes cutaneous candidiasis. It belongs to kingdom fungi. Um, Now, phytoconstituents have various therapeutic activities, but due to the drawbacks like poor solubility, low bioavailability, and here in case of limonin, it is volatile in nature. Its clinical uses are limited. So, to overcome these drawbacks, solid lipid nanoparticles offer a solution by encapsulating the bioactives um, in solid lipid core and stabilizing it by the surfactant, uh, due to which the stability is enhanced, absorption is enhanced, and it provides controlled release. So, moving forward to methodology, the solid lipid nanoparticles were first prepared and then they were incorporated into gel. So, for preparation of solid lipid nanoparticles, two lipids, glycerol monostearate, which act as a which forms the matrix, and it is a solid lipid was used, and second lipid that was stearic acid, which provides the hydrophobic nature, was used. So both of these lipids were first heated above their melting point, and for in case of aqueous phase, two surfactants that are tween 80 and span 60 were used. Both of them stabilize the enhance the stability, and tween 80 prevents aggregation while span 60 controls the particle size. Both of them were dissolved in water and distilled water and were heated at the same temperature as that of lipid phase. After heating, the aqueous phase was added to lipid phase under constant stirring to form a porous emulsion. Now, this porous emulsion, was homogenized at 800 bar for 10 minutes at 55 degrees Celsius and then cooled to form solid lipid nanoparticle dispersion. So, for the solid lipid nanoparticle formulation, we optimize the formulation using factorial design, for which the study type was randomized factorial, design type was 2-level factorial, 4 runs were done. Two factors that were selected were lipid concentration and surfactant concentration. Both of them were used in gram. The maximum value of lipid concentration was 7 and that of surfactant concentration was 3, and minimum value for lipid concentration was said to be 5 and for surfactant concentration it was 2. So based on this data, the optimized formulation was lipid concentration 5.590 gram and surfactant concentration 2.697 gram, for which the response for entrapment efficiency was 80.250% and the response for minimum particle size was 149.125 nanometer. Now, these optimized solid lipid nanoparticles were incorporated in gel by first dispersing the carbopol in distilled water under constant stirring and letting it swell for 1 to 2 hours. This swollen carbopol was neutralized using TEA, that is triethanolamine, and the clear gel was formed. To this clear gel, solid lipid nanoparticle dispersion, which we prepared earlier, was slowly added under gentle stirring to avoid nanoparticle breakage. A homogeneous gel was formed, which was ready for use. Moving forward to results, the quantification of limonin was done using UV spectrophotometer, for which the lambda max was found to be 280 nanometer, and from the linearity studies, R square was found to be 0.998. The particle size was analyzed, and the particle size were between 100 and 200 nanometer. The zeta potential was measured using ELS, that is electrophoretic light scattering, and it was found to be 4.2 millivolt. Next, the evaluation of gel was done, for which three batches were evaluated. All of them were colorless, transparent, uniform, smooth, and its odor was citrusy and somewhat sweet. There was no skin irritation observed in any of the batches, and for all the batches, viscosity was appropriate. So for batch 1 and 2, the pH was found to be 5.6 and homogeneity was good. And for batch 2, the pH was found to be 5.7 and homogeneity was very good. Moving forward to in vitro drug release study, it was done using transdiffusion cell apparatus. It was done for 24 hours and the collected samples were analyzed using UV spectrophotometer. Now, in the above diagram, we can see that the antifungal activity was evaluated, for which the first image shows that the antifungal activity of limonene at different concentrations. The concentrations used were 50 ppm, 100 ppm, and 150 ppm. Next is the placebo of solid lipid nanoparticles with no limonene, and the last one is limonene loaded solid lipid nanoparticles, where we can see a nice inhibition of the fungi growth. Next, we have used marketed gel, and in the next, it contains regular gel that contain only limonene. No solid lipid nanoparticles were incorporated into it. So, for conclusion, the limonene-loaded solid lipid nanoparticles provide novel and effective against Candida albicans infection. It also improves the stability, bioavailability, and its antifungal efficacy is enhanced. The controlled release of solid lipid nanoparticles reduce the toxicity and enhance the penetration. Also, the solid lipid nanoparticles protects the limonene from degradation, as limonene is volatile in nature, so it protects it from getting volatile, and it is safer and more targeted treatment option. These are some of my references. Thank you.
Thank you, Pranjali. So there are a couple of questions. So on what basis you selected limonene?
So limonene is a phytoconstituent, and based on the study, we found that limonene, there is no solid lipid nanoparticle or any formulation used that used limonene. So we used limonene for that.
But has anybody, or have you found any study where limonene is used in for the antifungal or for any skin disease?
Actually, limonene shows antifungal activity against Candida albicans, so that's why I used limonene. There are research articles present on the same.
So there is already study available.
Yeah, study is available.
Okay. Does the limonene penetrate in the skin?
If we use the limonene as such, as it is oil, it does not penetrate as much as if it is incorporated into the nanoparticles.
Okay, and why did you select it to prepare solid lipid nanoparticles?
The limonene is lipid. It is present in essential oils. So incorporating the lipid into the solid matrix helps better penetration.
So you are trying to penetrate that
Yeah,
in the skin.
yeah.
So have you looked at the any toxicity produced by this nanoparticle after penetrating in the skin?
I have not yet done the studies for toxicity.
Okay. And in your standard, the essay which you have done on the in the plate.
Yeah.
So was that also solid lipid nanoparticle as a standard?
The third picture shows where the limonene loaded solid lipid nanoparticles were used, and the middle picture shows the placebo, means there is no active compound in the nanoparticles.
But you also use the one standard, right? You
Yeah,
mentioned that gel, yeah. So
yeah.
that is also nanoparticle?
No, it is a V gel from Himalaya company.
So then how can you compare? Because the gel which you are using as a standard is not nano.
Yeah.
Right. Okay.
That's why I have used nanoparticle, because um, um,
That's fine. Thanks, Pranjali. It was nice presentation.
Hello.
yes, sir.
There are some questions from our audience. The first question is, how did the antifungal efficacy of the nanoparticles against Candida albicans compare with the pure limonene or standard antifungal drug?
It was done using the well plate method as shown in the pictures.
Okay, and which optimization technique can be applied to achieve stable and efficient solid lipid nanoparticles?
Sorry, sir, I did not get your question.
Which optimization technique can be applied to achieve stable and efficient solid lipid nanoparticles?
Sir, here I use the mean of the responses that I got.
Okay, and the same question similar to our Bade sir asked, that is, how does choice of lipid influence the efficiency of using limonene?
The solubility of limonene is lipid in the solubility of limonene is lipid, so the selected lipid showed promising formulations.
Okay, thank you, ma'am, for your clear and well-structured presentation. Your work demonstrates a flow nanotechnology and how nanotechnology can enhance the therapeutic potential of natural compounds. Thank you, ma'am.
Thank you, sir.
Now, move on for the presentation. Our sixth participant is Kalyani Ramprasad Tomre.
Yes, sir. Am I audible?
Yes.
Hello.
Yes, you are audible.
Can I
Now,
share the screen?
wait. I just introduce you and then you can share. Moving to the further presentation, the session is Miss Kalyani Ramprasad Tomre, a PhD scholar from SMT Kishoritai Boer College of Pharmacy, Campton, Nagpur. She will share the innovative research on 7O substitute tyrosine derivative as VEGFR2 inhibitors, a natural product based anti-cancer therapy. This advanced work conducted under the guidance of Dr. Krishna Gupta and Dr. Milind J. Umekar explores novel chrysin derivatives as potential VEGFR2 inhibitors, which hold great promises in targeted cancer therapy. Please join me in welcoming Ms. Kalyani Tomre. Now you can start, ma'am.
Thank you so much, sir. Sir, is screen visible?
Yes.
Okay. Thank you very much, sir. Myself, Kalyani Tomre. The topic of my today's research presentation is to Innovative 7 substituted chrysin derivatives as a VEGFR2 inhibitors, a natural product-based anti-cancer therapy. As we all know that cancer is a disease which is characterized by uncontrolled cell division and which is the second leading cause of a death in world at present. Number of scientists says that despite of number of advancement in cancer prevention, diagnosis and treatment, still there is a global burden of cancer death is drastically increasing. Hence, there is an essential need to develop anti-cancer drugs with maximum effectiveness and minimum side effect. It's a vital trend in anti-cancer drug development and research. As we all know, angiogenesis, which is a formation of blood vessels, every cancerous cells require oxygen and nutrition for their metastatic proliferation and growth. Hence, VGFR, vascular endothelial growth factor, which play a very important role in angiogenesis pathway. It exerts angiogenic effect by reacting with the VEGFR2 receptor, which is a play important role in angiogenesis pathway. So, and it is also found that number of cancerous studies reported that the concentration of VEGFR2 in cancerous cells is increases rather than in the normal cell. Hence, it is a vital competent target for the selectivity of a cancerous cells. So the inhibition of VEGFR2 can inhibit angiogenesis, and angiogenesis will ultimately inhibit the cancer process. There are a number of VEGFR2 inhibitors are available in market, such as sorafenib, pazopanib, etc. But their drug resistance and around 95% patient who are on the VEGFR2 inhibitor therapy possesses hypertension as a major side effect, followed by cardiac ischemia, which restrict their use. Hence, the development of new VEGFR2 inhibitor is essential. So in recent years, natural compound received much attention just because of their less side effect. Here we decide a flavonoid, flavon, which is a subclass of flavonoid, which consists of which has been found in various natural sources such as plants, vegetables, etc. It has been found that this flavonoids possesses a number of biological activity, including antihypertensive, including anti-cancer, and it has also potential to inhibit a VEGFR2 by introducing some other compound. So there is a SAR of chrysin, which is a flavone molecule. Chrysin consists of 3 rings, A, B, and C, where A ring contains 2 hydroxyl group, C ring contains oxygen and carbonyl group, and B is a aryl ring. So introduction of the substitution at 7 position can lead to the increasing anti-cancer activity. 5-hydroxyl group is essential for activity. Carbonyl group as well as this aryl ring is essential. And the substitution at any other position of that ring will leads to the decrease in anti-cancer activity. So on the basis of we further performed a number of in silico studies on different compounds. From this, we have found that ester and alkyl amine derivatives. Is screen visible, sir? Hello?
Yes, yes, visible. Screen is visible now.
It has been found that ester and alkyl derivatives shows a maximum activity, maximum safe study in silico study, that ester derivatives and alkyl amine derivatives, most importantly, they are easy for structural modification. Hence, we decided to substitute ester and alkyl amine derivatives at 7th position. So our rationale is to design a novel 7 substituted chrysin derivatives on the basis of VEGFR2 inhibitors. From the literature survey, we have found that almost all VEGFR2 inhibitors possesses 4 structural characteristics in their compound. First one is the head hinge region, second one is the hydrophobic region 1, third one is the HBM, that is hydrogen binding moieties, and fourth one is hydrophobic region 2. From this, we design the chrysin derivatives as shown in figure. That green line represent a pharmacophoric feature that will react with the glutamate 885 and aspartate 1046 in DFG motif of VEGFR2 receptor, which is a characteristics feature present in a VEGFR2 receptor. Then there is next hydrophobic region 2, that is again a distal aryl ring. And first one is there is a head hinge region, that is it may be a ester group, or it may be a aryl ring, or it may be a alkylamine ring. Then second part is a hydrophobic region 1. It contains a alkyl chain, which is mainly responsible for a lipophilic activity for the cell penetration. Hence, our aim to design and synthesize of a novel, potent, and efficient 7-alkylamine and ester substituted chrysin derivatives as a VEGFR2 inhibitors. For this, we firstly conducted a in silico drug study, followed by docking study on where we used a 4ASD PDBID of VEGFR2 inhibitor, followed by, on the basis of in silico study, we selected some derivatives for the synthesis process, then followed by characterization of the derivatives, and after the pharmacological screening of that synthesis derivatives as an antioxidant as well as an anti-cancer agent. In silico study, molecular docking were performed by using a software, molecular docking software, that is Pyrex, and again utilizing with the BioVia Discovery Studio. The validation was found to be, RMSD was found to be 1.2, which is a very good value we have found. Then ADME analysis, absorption, distribution, and metabolism and excretion. From this, we have found that all the derivatives follows the Lipinski rule of 5. It means drug likeness property possesses that derivatives. Toxicity analysis we have determined by using the software, that is Protox 3. And it has been found that all derivatives belongs to the category 5, which is very least harmful. After that, we go for synthesis. Here is a procedure for synthesis. The alkylamine derivatives were synthesized by incorporating chrysin with potassium carbonate in presence of DMF, dimethyl formamide solvent, stirring for 2 to 3 hours in presence of dibromoalkane. Alkane, it may be a dibromoethane or it may be a dibromopropane. We synthesized these derivatives, that is C1 to C6. In intermediate state, it is found that alkyl chain were introduced in the position of 7, followed by the reaction with various alkylamine derivatives at 80 degrees Celsius, and using the solvent DMF will generate a compound C1 to C6. The compounds are showing in figure. Then ester derivatives were synthesized by reacting chrysin with the various benzoate in presence of triethylamine solvent. The C7 and to C9 are ester derivatives. After synthesizing the derivatives to check the purity of a compound, melting point were determined by simply open capillary tube method, and it was found that all derivatives, almost derivatives, shows a sharp melting point. After that, the TLC were performed by using a silica gel as a stationary phase. Mobile phase were used acetic acid, ethyl acetate, and toluene in the ratio of 35:14:1. It has been found that all the retention factors were at a sharp spots, and sharp spots were visualized under a UV chamber at 254 nanometer. After that, we further proceed for antioxidant and anti-cancer activity, and simply antioxidant activity were confirmed by DPPH, free radical scavenging activity, from which order of potency are given in the table, that is C7 and C8. That is, the ester derivatives were found to be the most potent compound rather than other derivatives. And in anti-cancer activities, we have found that the 3 derivatives, that is C7, C8, and C5 derivatives, shows the major activity, maximum activity, rather than the standard compound, that is a sorafenib, is a well-known VEGFR2 inhibitor. So these 3 derivatives were found to be most active, and order of potency are given in the chart. And from this study, we have concluded that alkylamine and ester derivatives are synthesized as a VEGFR inhibitory activity to produce a maximum therapeutic effect. This finding have encouraged us to continue the synthesis of alkylamine and ester derivatives as a VEGFR2 inhibitors. Thank you. Hello.
Hello. Thank you, Kalyani. It was nice presentation.
Yes, sir.
So there are a couple of questions.
Yes, sir.
So you already decided the target, right? VEGFR2.
Yes, sir.
So
VEGFR2.
on what basis you decided?
Sir, VEGFR2 actually, at first aim was to that there is a no marketed drug which show a maximum effectiveness on the VEGFR2. We have found that the VEGFR2 inhibitors, which are already present in market, shows a number of side effects, and around 95% of suffering from this effect. and hence there is a no VGFR2 inhibitor present in the market. Hence, from this, we have decided to proceed with the VGFR2.
So there is no marketed product?
Yes, sir. There are number of marketed products. For example, sorafenib, pazopani, but their drug resistance as well as their side effect, most prominently hypertension, and which is followed by cardiac.
So how do you, in the future, if your molecule actually have the same issue, drug resistance, side effect, so how do you actually minimizing that at this level?
So to minimize this, we have chosen a natural compound. Such a natural compound which shows anti-cancer activity as well as anti-hypertensive activity, which is hypertension is the most prominent adverse effect of already presenting drugs. So we introduce such a molecule in a compound which shows the already anti-hypertensive activity, which can lead to the synergistic effect. For example, crysine. There are a number of literature available which shows that crysine is a well-known, number of studies are going on that crysine is a well-known anti-hypertensive agent also.
So are you trying to say that crysine 7 derivatives which you are synthesizing is natural compound?
No, sir. It is a modified.
Yeah. So once you modify it,
yes,
will
sir.
not it show any toxicity?
It may be shows a toxicity. For this, we have already performed a toxicity analysis by using Protox 3 software. We only decide such molecule which shows the minimum toxicity at in silico studies. Firstly, we are performing in silico studies to check toxicity, and then further we will go for synthesis.
Yeah, but you already mentioned that you also have done MTT assay.
Yes, sir.
So what was the concentration which was producing toxicity?
Sir, concentration was, there are 5 concentration from 10 microgram per ml to 100 microgram per ml. There are 5 concentrations.
So all showed toxicity?
All showed, no sir, but almost compound did not show the toxicity. Not all. For example, ester derivatives shows the minimum toxicity as well as the alkylamine derivatives.
Okay, so if you are saying that there are inhibitors, VGFR inhibitors, when you are trying to develop a cancer treatment, does the derivatives or molecules should have toxicity or, toxicity against the cancer cells, or should they actually help in the proliferation?
Sir, I didn't get you the question, so please repeat.
So you mentioned that your derivative is not showing any toxicity at the concentration.
Yes, sir.
So if it's not toxic, will it be useful in the treatment in the cancer?
Sir, for this, again, further studies required. That here we only perform the anti-cancer activity against MCF cell line. There are, again, further we go for VGFR2 kinase assay kit. It does not 100% confirm that these derivatives may ultimately 100% inhibit the VGFR2. It is a prediction. To perform this, we will go for VGFR2 kinase assay, then go for animal study also, and then again clinical trials number of then it may be possible that these derivatives may show or may be proven anti-cancer agent in a patient. This is a simple trial-based study which I have done.
Okay, have you looked at CAM assay?
Yes, sir, CAM assay, but we didn't perform it.
So can you do testing in the CAM assay in the chicken embryo to find if there are?
Yes, sir, we will definitely go for this.
Okay.
But sir, first target our VGFR2 assay followed by animal study.
Okay. Thank you.
Thank you, sir.
Hello. Okay, Kalyani, there are some questions from the audience.
Yes, sir.
Okay, the first question is, how did, that is your substitute modification of chyrosine enhance its binding affinity or specificity towards the VGFR2 compared to the parent compound?
Sir, actually VGFR2 inhibitors are mainly divided into 4 parts. That is ATP binding domain, alkyl chain, hydrophobic region 2, and pharmacophore. If only crysine, if I taken only crysine, then only 2 parameters satisfied it. But when we introducing other substituent like ester and alkylamine, it has been found that they satisfied all the four major characteristics of VEGFR2 inhibitors. And hence the docking score of chrysin is less as compared to the docking score of chrysin derivatives. So that chrysin derivatives ultimately inhibit the increases the binding affinity of chrysin.
Okay, the next question is, this derivative evaluated through only molecular docking or you use various in vitro or in vivo assays?
Yes, sir, I didn't get the question. Please repeat.
Where this derivative, that is a 7O substituted modification of chrysin, you do only for molecular docking alone or where in vitro or in vivo assays also performed to validate their anti-cancer potential?
Sir, firstly, we perform in silico. After synthesizing, we go for anti-cancer also. That is in vitro activity of anti-cancer. We evaluated the anti-cancer activity against the MCF cell line followed by the MTT assay. We perform in silico as well as we perform a in vitro anti-cancer activity.
Okay, the last question from the audience is, which in vitro cell models are suitable to anti-cancer activity performance?
Sir, MCF7, MDA, MB31 is again cell line. There are HC22T. There are a number of cell line available where we can evaluate the anti-cancer activity.
Okay. Thank you, Kalyani, for presenting such impactful and cutting-edge research. Your contribution highlights the importance of natural product-based drug discovery in modern oncology. Thank you. Now,
Thank you, sir.
okay, thank you. Now, the further section will be concluded by Komal Mandre.
Thank you,
Komal?
Shreyash. Yes,
Okay.
yes. Thank you, Shreyash. Can I audible, Shreyash?
Yes,
Hello.
yes.
Good afternoon, everyone. It's my pleasure to introduce our next poster presentation. The topic of the presentation is Pharmacovigilance and Real-World Evidence, guided by Professor Raghunath Pandurang Raut, and the presenter is Aishwarya Pingale. Let's welcome Aishwarya ma'am to share their work with us. Aishwarya ma'am, are you there?
Yes, I'm there.
Yes, ma'am.
Yeah, am I audible?
Yes, ma'am, you are audible.
Yeah. First of all, thank you everyone for giving me this chance to present here. And just a moment. Is my screen presented?
Yes, ma'am. Yes.
Okay, thank you. So, my topic is pharmacovigilance and real world evidence. Actually, we, as in pharmacy profession, we emphasize on the research work, and we are researching on various topics, various drugs, and after the research, there are various steps to take in front or to take the drug into the market. So pharmacovigilance is the one of the stage that will help us to provide our drug into the market, where the pharmacovigilance, it provides the safety and effectiveness of the medicines. There, for this, there are various stages in the pharmacovigilance that we take while conducting the drug trials, and that trial stages coordinates with the pharmacovigilance, that it is putting an eye on the drug. And also we get the feedbacks from the drug and the person who is consuming the drug, so that data is totally combined into the one of the electronic device or one of the electronic software that gives us the effectiveness of the medicines. That is nothing but the real world evidence, that we keep an eye also with the evidence that our drug is very effective on the patient that we are giving. So before that, we need to do research on a drug. Well, we bring up the new drug, and after bringing up the new drug, do we do the trials on small scale, then large scale, and then into the market. And after coming into the market, there is some stages that we are keeping the eye on it. We are taking total feedback from the patients who are consuming our drug. So whenever we are doing the research, we should also take care that this drug should be also come into the market, whichever drug we are researching, because that gives a total impact of our work that is fulfilled by it. Next, the introduction part of the pharmacovigilance and the real life evidence is that the pharmacovigilance plays a very vital role in ensuring the safety and effectiveness of the medicines. That is most important. And next, the real life evidence is generated from the data source of such as the electronic records, patient registers, or the spontaneous reporting from the patient reports or the outcomes. So there are the various steps where we can take the outcomes and perform our studies and improvise our drug according to the market or according to the effectiveness of the drug. So after getting that data, we can improvise our drug. So there are various methods where we can extract the data or extract the stages of the drugs. That is the data source that we get from the spontaneous reporting or the EHRs. And after that, the collection of data, then signal detection and assessment, where we do the assesses and the causality clinical relevance through the accepted review, and then the understanding and interpretation, that what we can make the changes in the drug to improvise them and be our drug into the market. Next is the AI in pharmacovigilance and the real world evidence. AI is now playing a crucial role in our each and every part of the study related or the um, other related substances also. So AI is emphasizing on the pharmacovigilance also, where the anything we ask to the AI is giving us the answer. But ai is from this AI is also getting the information from the person only. So AI is the references of the person that we are taking the results from them. And next, whenever we are coming to the real world evidence, it supports risk detection fasterly and regulating the decision uh, of the decisions and making the medicine very safer to get consumed. So this is the theoretical part which we have come forward with the pharmacovigilance and real world evidence, because whenever we are formulating the drugs, we should also take that which formulated drug into the market. So we are now studying the further phases to bring our drug in the market, and also we are doing some research work on the various drugs. Recently, we have done the research work on the herbal hair oil, and we have uh, also published that research. But now we are taking this research work into the market levels, and we are researching that how we can take our drug into the market. So, thank you. This was the presentation.
Thank you, Aishwarya, for the presentation on this pharmacovigilance and real-world evidences. There are a couple of questions from my side. So you mentioned that you done a formulation of a hair
Yes.
oil. So
Yes.
relevant to that, can you tell me that how the pharmacovigilance of herbal formulation is done in India or anywhere in the world?
Yes, sir. The pharmacovigilance is the last stage or the fourth stage of the clinical trial, we can say. And after the fourth stage of the clinical trial, the pharmacovigilance mainly plays its roles. First of all, whenever we are introducing any of the drug or any of the formulation of the drug, we are first of all experimenting it on animals and then on the peoples. Then we take a couple of peoples first, and if it passes from the couple of people, then it carry forwards to the group of people and then into the market. The pharmacovigilance now plays its main role when it is in the market and collecting the data from the market level and showing the safety of the drug.
My question is that how the reporting is done for herbal.
Sir, yeah, reporting we can take from the patients which are using our hair oil. Secondly, the medical level who are giving the or selling the formulation to the patient, that they can report us. Thirdly, taking taking the reports from the EHR which we have created for the herbal oil, and then we can make a proper report that yes, our herbal oil is working properly.
So what is pharmacovigilance?
Pharmacovigilance, in my perspective, is nothing but the eye which we are keeping on our drug to make the safety that and effective, to see that our drug is very safe and effective towards the patient which we are using that.
So it is regarding the adverse effect.
Yes.
It is regarding the reporting, right? So how does the user come to know that there is an adverse effect of the hair oil or herb?
Yes, the user can come to know the its adverse effect from when the member or the patient is using the herbal oil. First of all, the herbal oil is working properly, then he will have the growth of the hair or and have the good effects. But if he is sensitive or he is having the any of the reaction from the agent that we have used in the herbal oil, either there will be the hair fall, either there will be the scalp irritating. So from that we can know that he is having the adverse effect of our drug, so this can be reported.
How is the ADR reporting done in India?
Sir, it's done through various ways, but most of the way which we are using is that spontaneous response from the patients. So that we have the particular batch number. Whenever we are manufacturing, we have the batch number. So the medical person, when he is taking the complaint from the patient, he writes down the batch number, that this batch number is having particular this effect, and then we can stop that production or check whatever wrong is going in that particular batch, or we can then check and come that back with our formulation if it needs the any changes in it.
Okay, thank you.
Thank you, sir.
Ma'am, we will take one more few questions. How can real world is in detecting rare or long term adverse drug reactions?
Sorry, ma'am,
Hello,
you
ma'am.
are not audible. Yeah.
How can real world evidence complement traditional pharmacovigilance systems in detecting rare or long term adverse drug reaction?
Actually, pharmacovigilance and real world evidence can go hand in hand whenever we are uh, taking the drug into the market, because the real world evidence is nothing but having the documentation of the drugs which we are coming up with it. So according, if we are thinking about the long term effect, of course it can help us in long term effect, because we are having the proper record of the effects and the adverse effects which is caused by the drugs.
Ma'am, second question is, what are the major challenges in integrating real world data from the electronic health records?
Integration is one of the part where the member is there to take the data from the data source and then putting up into the EHR and then integrating to the uh, software which it is been needed. But this is the thing where the complication can happen, or the things that can go wrong. But if we are taking it directly without integrating, the data source is directly connected to the EHR, and we can directly have the documentation, no need of the integration from the other sources.
Okay, ma'am, there is one more question. How are casualty assessment methods applied in pharmacovigilance? Hello, ma'am.
Hello.
Ma'am, how are casualty assessment methods applied in pharmacovigilance?
The casualty assessment we can take, such as stopping the manufacturing of the drug or the particular batch, and then improvising the drug. Secondly, first it firstly when we are coming up with the clinical trials, we can check there where we are going wrong, or where the modification is required. There then we can take our assessment and the protection that our drug is safer and gives it the perfect result that we are assuring from it.
Okay, ma'am, thank you for the insightful presentations. Let's now move to our next poster presentation. The topic is in silico identification and evaluation of bioactive plant product constituents of Glycyrrhiza inflata using structure-based drug design and ADMET analysis on HSV glycoprotein B, guided by Sachin Sharma and Krupa Badwa, and the presenter is Vishwakarma Rakesh Kumar. Let's welcome Vishwakarma for their presentation. Are you there, Vishwakarma?
Hello, ma'am.
Hello?
Yes, ma'am. May I present myself?
Yes.
Is it visible?
Yes, ma'am.
Hello, everyone. Respected teachers, sir, and all my friends. Good afternoon to all. I'm going to present my poster, and my topic is in silico identification and evaluation of bioactive plant product and constituents of the Glycyrrhiza inflata using structure-based drug design and adiabatic analysis on HSV glycoprotein T, under guided by Sachin Sharma sir and Krupa Badam ma'am. Now, first, let's start with the introduction of the virus. So, virus is the microorganism species. Again, viruses are the obligate intracellular parasite. It means they replicate or multiply inside living host cells. So, particularly focus HSV simplex herpes virus, which is also known as herpes, is a common infection that causes the painful blister or ulcer. It means uh, particularly shows the skin to skin contact. You can say directly transmission. So, majorly includes the herpes simplex viruses HSV1 and HSV2. The common pathogenic that remain a serious threat to human health. It means that they produce the disease to the humans. So particularly focus on the glycoprotein T, which is a very important and unique component of the herpes simplex viruses. Envelope which is essential for the virus entry by binding host cell receptors, just like nicotinic nectin 1. So particularly if I am asking about the nectin 1, is the cell adhesion molecule especially present of the epithelial and neural tissue. Again, next is the HBEM. The HBEM is the herpes virus entry mediator. It is a protein and co-receptor of the herpes viruses, and it is the tumor necrosis factor receptor subfamily. And another one is the 3-O sulfated herpes. Next is the scientific classification, and this is a morphological classification of the HSV virus. So, particularly asking about the nucleic acids, the genomic information is in DNA, and it is in linear DNA for. Next is the arrangement of the nucleic acid, which is in the double stranded. Next, another one is the envelope. Envelope is a particularly HSP virus is a lipid bilayer and derived from the host cell membrane. Studied with the viral glycoproteins, it means GB, GC, GD, GH, GH, essential for the host cell entry. Next, it is symmetry, ecohedral, and family is the Herpaviridae. But particularly, if I am asking about the family Herpes viridae, so it is again divided into the subfamily. So subfamily includes alpha, beta, and gamma. HSV 1 and 2 comes under into the alpha herpes viruses. Next is a plant product. The major medicinal and bioactive plant products derived from the natural. So one of which the Glycorrhizia inflata. To so its family is Fabaceae and subfamily is again Fabaceae. It means it has a family of the flowering plant. It's Latin, but faba means bean. Next is genus Glycyrrhizia. Glycyrrhizia is a Chinese plant species. The genus is Glycyrrhizia. Is a more likely licorice species one found in traditional Chinese medicine. The plant chemical constituent are the glycoprotein, glycochalcone A, isoquercetin, as well as glycyrrhizia isolated, which is already shown. So first one is the glycopolymeric. The main nucleus is present in the glycycoumarine is the benzopyrrole or coumarine. It is a derivative of the coumarine. It is a fused of the benzene and alpha pyrrole. Or alpha pyrrole is also called unsaturated electron group. And another side chain is the pyrene group. Next is the licochalcone A. So licochalcone A is a retrochalcone flavonoid, which is derived from the Glycyrrhizia species licorice root. It is a derivative of the phenochalcone. Next is the isoliquidine. So isoliquidine is the monosaccharide derivative that is a trans-chalcone and substituted by the OH group at the 4 and 2nd position. Next is the glycoresin. This is glycoresin, which is also known as glycyrrhone or glycoresin. It is a triterpenoid saponin. It is a glycoside derivative. It a tricarboxylic acid, pentacyclic triterpenoid, and conjugate acid of glycoside. Next is the liquid. Liquidity is a flavonol glycoside that is a liquidization attached to the beta-D glycopyranosyl residue at 4th position by a glycosidic linkage. Next is my objectives to characterize bioactive constituent of the glycogen beta using phytochemical constituent, analyze and validate 3D structure of the HSV and target glycoprotein D for molecular docking stimulation and amino acid interaction we will see later. This is the pharmacokinetic properties involves absorption, distribution, ADMET, or the drug likeness evaluation. Toxicity prediction and drug likeness of the promising compound using in silico tools such as Swiss ADMET products 0. To compare the computational performance to the Glycyrrhizia inflata constitute with the HSV inhibitor acyclovir, which is my standard drug focusing on the binding affinity.
2 minutes remaining.
So particularly all my 5 chemical constituents I compare with my standard drug acyclovir. Next is the methodology. In the methodology, first of all, I discuss about present molecular docking stimulation. First one is the protein preparation. So my organism is the antiviral, which and my target is glycoprotein D. The 3D structure of the following target protein D, PDB ID is a 5CBA, was taken from the protein bank database. Their website is www.rcbs.org. Investigating the investigate the binding affinity of the GD for its surface receptor, performed a knowledge-based protein ligand docking. Next step is the ligand preparation. So this the particularly ligand preparation involves draw the 2D structure, its energy minimization. So first one is the camera structure used for the particular 2D structure preparation. Next is the Chem3D for the energy minimization. It is used for the give the stable conformation of the ligand. Next is a Swiss ADMET, which is gives the uh, drug likeness properties and Protox
Time up.
3.0, which toxicity prediction. Next is Autodoc Vina, which is prominently used for the uh, preparation of protein and ligand. And discovery study BioB is a visualization of the prepared ligand and receptor. These are the amino acid interaction. Start this is one is my standard, and another 5 is my plant
Time up.
constituents. So particularly I am so all of this M5 compound minus 3.8 gives the best binding affinity. Next one is the biological ADMET evaluation. I use ADMET evaluation, particular Swiss ADMET. So it follows the lipid glucose determine the drug likeness property of the Swiss ADMET use. Next is the Protox in silico toxicity study, which I Protox 3.0. This is the link, and my standard drug shows the carcinogenicity, which will cause the cancers and another plant constituent or are inactive except immunotoxicity. Next is the biologically prediction. I use the mole inspiration. This is the link www.molesinspiration.com, and you can say revalidation of the drug likeness properties. This is my conclusion. All of the I use the in silico prediction for the in which the one of the best show the M5 display the favorable docking score, and its binding affinity is the minus 3.8.3 kilocalorie per mole. This is my references.
Hello. Hello.
Hello.
Vishwakarma ma'am, thank you for the presentation. It was nice presentation. What are you going to use as your in vivo model to confirm your results?
But particularly I focused in silico work if it is possible, so I will confirm with in vitro performance.
Sorry?
you repeat the question?
How will you confirm or what in vivo model you will be using to confirm your results? Hello.
Yes, sir.
Okay, so don't worry. What tool you have used for molecular docking?
Sir, Autodoc, Vina, Discovery Studio, Biovia, and
So what is the purpose of Discovery Studio?
visualization and preparation of the ligand and protein.
Okay, and the molecules, you have only taken 5 molecules from the plant? Or there was so 5 molecules you have taken?
Yes, sir. I have taken major 5 active molecules because as per I validate the validate according to molecular doctrine.
Okay, thank you. Thank you.
Hello, ma'am. There are some questions from audience for you. First is, how does resistance to acyclovir develop in herpes viruses, and what strategies are used to overcome this resistance? Hello, ma'am.
Hello. Can you repeat this question?
How does resistance to acyclovir develop in herpes viruses, and what strategies are used to overcome this resistance?
So, particular, if I am asking about the acyclovir, I take as a standard because the glycoprotein D, there is no standard drug and every approved drug is available. So, there is first I studied the structure activity relationship and there is a possible conformation changes. So may I improve the resistance of the setting.
Okay, ma'am. There is one more question. How can findings from this in silico study guide future lead optimization and semi-synthetic derivative design of Glycyrrhiza inflata constituent? How can findings from this in silico study guide future lead optimization and semi-synthetic derivative design of Glycyrrhiza inflata constituent?
So particularly Glycyrrhiza inflata is not only one, but there is a lots of the Glycyrrhiza species. There are the available as a Chinese plant, which is again available for the Licochalcone A. Another one is Licochalcone B, C, D, or we can use more than more, more than 50 chemical constituents I will use for the virtual screens.
Okay, ma'am. Thank you. Thank you for the insightful presentation. I hope you are enjoying the conference so far. We are now moving on to our next poster presentation. The topic of this presentation is green synthesis and antimicrobial activity of bispyrazole, guided by Dr. Machindra J. Chauhan, and the presenter is Dr. Vishal More sir. Let's welcome to Vishal sir to share their work with us. Vishal sir, are you there?
Yes, ma'am. Am I audible?
Yes. Yes, sir.
Good afternoon, everybody. Okay, I'm going to share my poster. Is it visible, ma'am, the poster? Hello?
Yes, sir. Yes, sir.
Okay. So the topic of my poster presentation is green synthesis and antimicrobial activity of bispyrazoles. Here, green synthesis, I mean to say I have used microwave assisted synthesis for the second step here. I have used both the methods, conventional synthesis as well as micro-assisted synthesis. Therefore, I have named the synthesis as green synthesis. So pyrazolines are the compounds which are the important heterocyclic compounds and having the array of biological activities like antimicrobial in that antibacterial, antifungal, anti-inflammatory and analgesic properties. Therefore, the pyrazoles have better future in the synthesis as well as in pharmacy. So I have synthesized the bis pyrazoles and I have conducted the antimicrobial activity, that is antifungal and antibacterial in that. So the methodology of synthesis, I am going to my scheme here. I have used ethyl acetoacetate. Ethyl acetoacetate was taken in conical flask and mixture of ethanol and hydrogen hydrate is dropwise added. With the stirring, I got 5 means I got 3-methyl pyrazole 5-one. That is the heterocycle I got here. And then I have used this heterocycle in presence of aldehyde and the solvent used was DMF. And this second step I have done with the micro assisted synthesis. I have added 2,4-dinitrophenylhydrazine and I got this fourth position, I got the aldehyde substitution, and second position, I got 2,4-dinitrophenyl substituted. So here I got the pyrazole, that is bispyrazole. In third step, the chloroacetic acid was added along with the ethanol, and the free amine present at the first position of acid group on the first position of that nitrogen. Okay. Then in the next step, I have used orthophenylene diamine and ethanol. So the last step is means the synthesis of benzimidazole is the reported in Ogle's practical organic chemistry. So I have prepared the benzimidazole nucleus at the first position, and hence I got the bispyrazoles along with the benzimidazole molecule. So here I have synthesized 8 derivatives, 3A to 3A to B, 5A to B, 3A to 3F, then 5A to 5B. So these, according to this R1, R2, and R3, I have taken the different aldehydes. The micro-assisted synthesis, for micro-assisted synthesis, I use catalyst Kata R, 32 liter and 850 watt. This is the instrument used for micro-assisted synthesis. And biological activity I have carried out along with the standard drugs. I have compared the biological activity, that is antimicrobial activity and antifungal activity. I have compared with the standard drugs. For antimicrobial, antibacterial activity, I use amoxicillin clavulanic acid for gram-positive bacteria, cefixime for gram-negative bacteria, and ketoconazole used standard for fungal infections to check the activity of fungi. Okay. The characterization of the synthesized compound is done by IR, NMR, as well as elemental detection. And the synthesized compounds yield is in between 67 to 80%. I got it. And new chemical entities containing pyrazolines and heterocyclic acids, that is pyrazole and benzimidazole, are synthesized using conventional as well as microwave assisted synthesis methods. And several compounds showed notable antibacterial and antifungal activities. So here I have used the bacterial strains as Streptococcus mutans, then yes. So compound 3A, 3B were notably active against Streptococcus mutans. Compound 3B also showed moderate antifungal activity. Then compound 3C had minimal activity, while compound 3D and 3E showed moderate activity against selected bacteria and fungi. And compound 5A and 5B showed good activity against Streptococcus mutans. And a future scope for this, I am going to synthesize more derivative by adding different aldehydes, as well as want to synthesize the compounds after substituting the amino position at the benzimidazole. So this was about my presentation. Thank you, ma'am.
Thank you, sir. It was nice presentation. Just a couple of questions from my side.
Yes.
What was the yield quantity?
For minimum yield, I got 67% and maximum 80%. So detailed table I haven't put here. That is along with me.
Okay, and what was the concentration you use, which have shown activity? So with the compounds, you mentioning about antibacterial and antifungal. So what was the concentration range?
Sir, I have taken 1 mg per ml concentration for that.
1 mg. Okay. Okay, sir. Thank you. Thank you from my side.
Thank you, sir.
Hello, sir.
Yes, ma'am.
There are some questions from audience.
Yes.
Which biological targets are most commonly modulated by pyrazole derivatives, and how does substitution on the ring influence their activity? Which biological target are most... Yes.
Madam, your voice is breaking. Hello.
Hello, sir.
Hi. Yes.
Hello.
Hello.
Which biological target are most commonly modulated by pyrazole derivative, and how does substitution on the ring influence their activity?
Substitution. The second part of the question, I am going to give the answer. So ring substitutions means if there is a there are bulkier groups. Chloro 2,4-dichloro. Here I have taken chloro and 2,4-dichloro. So 2,4-dichloro and chloro derivatives are more potent than the other derivatives.
Okay, sir. One more question. How does bispyrazole compare in antimicrobial potency with standard antibiotic?
Yes. Pardon?
How do bispyrazole compare in antimicrobial potency with standard antibiotic?
Last word I am not getting. Which antibiotics?
Antibiotic. Standard
Antibiotics. How do bispyrazoles compare in microbial potency with standard antibiotics?
Yes.
Frankly speaking, any chemical is giving the antimicrobial activity. These derivatives are what is I got here. These are not potent compounds, but they are still having the antimicrobial activity.
Okay, sir. Thank you, Vishal sir, for the wonderful presentation. It was very informative.
Yes. Yes.
Thank you to everyone for your attention and for actively listening during the conference. We appreciate your engagement and participation. We look forward to having you with us for the upcoming session as well. There will be short break. After the short break, we will have a session on the genetic biomarkers and fish in accelerating drug discovery by Dr. Enrico. Sir joining from Italy. And this part hand over to Sanjana and Shri. Please join us at 2:30 p.m. for the session. Thank you so much.
Thanks a lot, ma'am. It was very nice.
Everyone, please join in 2:30 for the next session. Thank you.
Hello, good afternoon.
Hello.
Hi, good afternoon, Enrico. How are you?
Fine, thanks. How are you?
Yeah, fine, thanks. How are you? Sanjana, Shri.
Yes, sir.
Yeah, we can start now.
Okay, sir. So, welcome back, everyone. We now move into a highly anticipated session of the day. The topic is genetic biomarker and fish, accelerated drug discovery, a field that is transforming how we understand disease at the diseases at a molecular level. Genetic biomarkers are key in identifying disease risk, predicting drug response, and ensuring personalized treatment. FISH, or fluorescence in situ hybridization, is one of the most powerful tools in molecular biology that helps detect and localize specific DNA sequence on chromosome. Together, they are revolutionizing cancer diagnostic and drug development. uh, Taking ahead to enlightening us on the fascinating subject, we are honored to have with us Dr. Enrico Di Otto. He is a biologist with a PhD in oncology and specialization in clinical pathology from the School of Medicine, Bologna University. Dr. Enrico is the CEO and co-founder of OACP, a medical biotech company pioneering rapid molecular diagnostic for cancer. With more than 20 published papers in his credit, he has made a remarkable contribution to oncology research. He is also the inventor of an innovative technology that improves the efficiency of nucleic acid hybridization, a true game changer in molecular diagnostic. With such an outstanding profile and expertise, we couldn't have asked for a better speaker for this session. Please join us in giving a warm virtual welcome to Dr. Enrico Di Otto. Sir, this stage is yours now.
Thank you very much for the introduction. Let me starting sharing my screen. Okay. I hope you can see my screen right now. And yeah, I will go this speech, this presentation. After the presentation, I can share eventually the PDF with the organization, so you can have it on your end available together with my contact. And if you have any questions, feel free to ask, mainly by chat or just raising hands, and we can try to address that. So, as perfectly introduced, today I will address with you the role of genetic biomarkers and in particular fish, so fluorescent in situ hybridization, in accelerating drug discovery. So I will just introduce a little bit biomarkers, the rationale behind the usage of biomarkers, and then quickly fish and a couple of examples. So I will let you, a starting point to go deeper in the in the argument in the next days. So biomarkers, as you know, are widely used since probably hundreds of years. They can be serum biomarkers, they can be DNA biomarkers, they can be they call informatic or virtual biomarkers. They are used for immunotherapy, transplantations, target therapy, surgery, and so on. So starting from the definition of biomarkers, so basically they are biological indicators of normal or disease processes that are in in the in your in the body of patients. As I say, they can be measured in different part of the body, so blood, tissue, fluids, and so on. Most important thing that commonly, especially in our when I studied biology, but mainly clinical pathology, we were commonly brought to think about biomarkers like just for pathological states, but they are probably more informative to check the physiological states and the physiological conditions of a patient. And without knowing how biomarkers, are in a healthy during the healthy phase of patients, or when patients still be so active in the interpretation of a pathological biomarker. So, seeing that biomarkers that reinforce the fact of the usage of biomarkers in drug discoveries, because we must be very, very aware on how biomarkers ranges between healthy populations to be able to effectively address a medical need, Right now, biomarkers are the key of clinical trial designs and patient selection. Mainly what I can, going deeper, so avoiding the proteins, blood proteins, and so on. Let's go to genetic biomarkers. We are talking about DNA or RNA eventually, and they can be related to mutation, fusions, polymorphisms, expression levels, why not? And by testing those biomarkers, we can identify how pathology, how diseases are progressing or are responding to treatments. Think about oncology is the best example of that. And they can be, as mentioned, and as you can imagine, diagnostic, so like BCR able, prognostic or predictive, like HER2 in breast cancer. So they are prognostic because having a HER2 positive breast cancer means a more aggressive type of breast cancer, but they are HER2 positivity is predictive of a potentially good outcome if the patient is treated with target therapy, monitoring safety, and so on. And as I already said, there are multiple roles, so it's not a one-to-one relationship. So biomarkers, while the discovery is progressing, is easy to find one marker that can have multiple functions, especially in patient management and monitoring. The most important in drug development are obviously the predictive and prognostic ones, because a predictive biomarker is related with the way patient respond to a specific therapy, or if not treated at all. And to my approach, it is the most important point. So the baseline approach to me, and this is my experience, and happy to share your thought with me as well. To me, the level must be first of all, it's better to know the physiological state of biomarkers we are interested in, especially in the population, especially in the variability of the population where people came from, and then go deeper into those biomarkers behaviors if patients are not treated. I know obviously it's something that can be done retrospectively, mainly because I don't see any ethic committee that can, approve a study where you have a patient with a clinical condition and you state that you will never treat the patient for the clinical condition. So, but it is important because by knowing how predictive biomarker in a specific condition evolve and how the condition evolve together without any interaction from outside or specific interaction from outside. Maybe you can use murine models to study that. And then the response of that biomarker when addressed with a specific therapy, like ALK fusion. It was a turning point in aggressive lung cancer treatment. The crizotinib, for instance, is the first treatment for ALK as ALK inhibitor. And then prognostic markers is an average, let's say, is what I say. So how the disease evolves without regardless treatments. And then, those are two classes that are related to companion diagnostics. Basically, again, genetic markers can be very, very specific and, they are starting from the discovery of a mutation, of a specific mutation that can support you as a researcher eventually in addressing the clinical condition that harbor that mutation. In that case, I show you a key example, which is the BCR-ABL fusion, which is a driving mutation for CML. And obviously, all the efforts were made on that, to address that mutation, to try to find a very specific drug or treatment for the CML. Obviously, when you target a potential, biomarker earlier, it is involved in the, the clinical condition developments, better will be the outcome for patient if you can find a drug or something that can block the effect of that biomarker during the pathology, let's say. And yeah, obviously, one biomarker sometimes is not enough because tumors, especially in oncology, tumors develop genetic resistance mechanism. As I was saying, as ALK, as first biomarker we introduced for lung cancer, there are tumors that developing resistance to ALK inhibitors by promoting the amplification of HER2 gene, like breast tumor. So this is why I stressed a little bit on how. So you need to know how biomarkers evolves, which is their function, and so on. Because sometimes you need to think on horizontal and not vertical while studying pathology or discovering new biomarkers or new drug for specific conditions. One of the key points that probably make the difference and is making the difference in the drug discovery is the graph that you can see on my left part of the screen. So, when you have a biomarker that guide, or more than one biomarker, for instance, but let's say one biomarker that guide your clinical trial, statistically, it is easier to find a very nice outcome for the study itself by increasing the potential, the possibility of succeeding in proving that your drug is effective on patients. And it is not just only to have a nice study published, but it is for the implications on that there will be for patients, for instance. So think about this is a graph from the clinical study for crizotinib approval. And so they only, enrolled ALK positive lung cancer patients and tested them with ALK inhibitor and without ALK inhibitor. So they didn't take all the non-small cell lung cancer patient at stage 3 with metastasis, blah, blah, blah. They just tested this non-small cell lung cancer patients, tested for ALK rearrangement, positive, okay, let's go to the trial. And you can see how the median progression-free survival rate is differentiated between the case and the control cohort. And that means, from a practical point of view, because clinical studies cost a lot, so when you are developing, so you know probably better than me, but so clinical studies have different phases. So you have phase 1, which is the preclinical, and phase 2, the 1 and 2 are the, let's say, preclinical, phase 3, the clinical level, then you have the the post-clinical monitoring. And if you're starting your journey, your trip from the drug discovery to the drug approval, totally blind, so without any biomarkers, but just clustering patients and populations for the type of condition are you try targeting, let's say lung cancer. So just to keep coordinates with the previous slides, the potential of success is way lower to pass from phase 1 to phase 2, and then it is lower to go from the phase 1 to approval. So if you go totally blind, just non-small cell lung cancer, stage 4, node metastasis, resistant to standard treatment, you may have an 8.5% of success rate to have your drug on the market. But if you overall start selecting by using biomarkers, so you can say non-small cell lung cancer, stage 4 patients with resistance to standard treatments that are ALK positive, you may have 25.9% of success potential, probability of success, to have your drug from your laboratory to the market. On the other hand, that means that if you are going to fundraise your R&D because you are a startup, or you have a grant proposal, or you want to go to a charity to get your project financed, you can display that slide and say, okay, we identified that 1, 2, 3 biomarkers to very to make a funnel of patients in a very straightforward way. So we maximize the probability of success. That means that we will maximize the probability of return of the investment if it is a startup, or benefit on the patient, future benefits for patients if it is a charity or a project, whatever. So biomarkers are key, not only for current available drugs or treatments, but also when you are going to discover a new drug. So if you are a researcher in discovering biomarkers, it is the same the same value that you are bringing. So start thinking about not only of the biomarker by itself, but think about the biomarkers you are going to discover through total shotgun NGS or whatever you're doing, that as much specific the biomarker is, as much useful it will be. Last but not least, then for the first part of that speech, the regulatory impact. So that's probably it is more for industry. It is a pain for industries more than for probably R&D. You have difficulties in maybe ethical committee, finding partners, select patients, and so on. When you are going to the industry, probably, at least for me, that I'm in the diagnostic industry, before starting something, I go straightforward to the regulatory, part of the project. this also if it is just on a scratch, so that we are starting a project from the scratch. The first point is not how many patients, which is the fine tuning of the technology. The first point I'm going to check is the regulatory landscape or framework in which my diagnostic product will fall into. And the same, obviously, if it is a drug, sometimes it is clearer where the drug is boxed in. But so, if you have biomarkers, it is easier for regulators to approve your drug for medical use. Especially thinking about that more than half of recent drug approvals utilize biomarkers data. Personalized medicine is well, accepted, and it is the final point of the current medicine. But looking current, it personalized medicine. They are starting applying it since probably early 2000. So probably earlier. And as you can see, you maybe find that there are guidelines to streamline biomarker validation and companion diagnostics co-development. So because of the benefit that I mentioned. And that's the first introduction on biomarkers. So there are a lot of technologies or to discover biomarkers. You can use whatever you want, basically. Mainly right now, NGS, it is used because the cost is is going dropping down since it was in place. NGS is a very high amount of information providing tool. So, but probably there is too much. If you sequence you made the whole genome sequencing of also let's start a rare disease that go to the whole genome sequencing, you may find x hundreds of mutations that mainly make no sense at all. In the past, we use more the array CZI, that they are very good to screen mutations as well. But I worked in fluorescent in situ hybridization, obviously, so I am a little bit biased on that. But all those high throughput, high output techniques need at a certain point for you to be able to localize exactly where the mutation is located, is collocated in the sample. So in which cell the mutation is present, how many cell of your tumor brings that mutation, also maintaining the spatial and the geographical order of the geographical setup of the cell, and how tumor cells interact with normal tissue cells, and if those mutation is more present on the on the connection between the tumor tissue and the normal tissue, or is inside the tumor tissue, if it is in the in situ components of the tumor, or if it is in the invasive component of the tumor, if the tumor is made of those two components, and so on. So this is the superpower of the fluorescent in situ hybridization technique, because FISH allows you to visualize directly within the cell using a fluorescently labeled probe. Basically, it like to tag the cell for the specific gene or genetic alteration or chromosomal alteration you are looking for. The probe principle is easy. So you have a probe that is designed to be complementary with the nucleic acid strand sequence you are looking for, and that complementary probe is directly labeled with the fluorescent molecule. So you then go to a procedure to properly manage sample or prepare sample, hybridize the probe, wash, and then when you're going to watch it, the probe spots remain on the target location. And as I say, it preserves the spatial context. Signal appear in intact cell or chromosome spread if you have chromosome, and so you can easily count the amount of those alteration, the cell that brings alteration or not, and so on. The funny part of the fish is that you can easily use different type of specimens. So you can have formalin fixed, paraffin embedded samples, chromosome spreads, cells, biopsies, cytological smears, cell blocks. Cell blocks are when you have a cytology sample, you collect cells that are in a liquid sample and then put in a formalin block. Probes are pretty big DNA or synthetic DNA fragments, about 200 kilobase. Think about primers for PCR that are 16, 20, 30 base pair. They are kilobase. And so that size allow you to target genes or regions that for sure, which deletion or example amplification for sure brings a clinical impact. To give you an example, in the past for brain tumors, we used to test the deletion on a certain region of the chromosome 1 and on the chromosome 19. The first way to detect those deletions was through Singer sequencing with a technology that it is called loss of heterozygosis. So they're going to sequence the two chromosome 1 and two chromosome 19, and if there is an heterogeneous deletion, you can detect it by checking the difference in the signal. They discovered that by detecting one base deletion makes no sense from a clinical impact point of view. So they started using fish, and so they discovered that by detecting 200 kilobase deleted in one chromosome, there is a better correlation with for the biomarker to be informative. So now 1P deletion and 19Q deletion for fish is diagnostic for oligodendroglioma versus astrocytic lesion or other lesions. That means a lot. So oncologists at some hospitals need just the 1P 19Q deletion with fish as a diagnostic marker, and then keep the histological examination and parameter for a second, to fine, to tuning the diagnostic report. But the key is that fish test. This is why I'm not aware of having a very big probe. You have also probes that are 1.2 megabase, so way bigger than that. The turnaround time fish can vary between the standard that is 2-3 working days. We as a company develop a rapid buffer that allow users, laboratories to make fish test in 2 hours or flexibility with a certain level of flexibility. So that means in the same day, you may speed up your R&D, your discovery, your diagnosis, and so on. As you can see in that image, I cannot find anyone for it. It missed a little bit of resolution in transferring from PPT to PDF. Apologize for that, but eventually I can send more. So this is how a fish test look like. The blue is the chromatin, generic chromatin, so they are nuclei in blue. And then you have red and green signal dots that are one red, each signal is one probe that hybridized the counterpart. So what laboratories do. I see the question. I reply just in a couple of minutes. What laboratories do is just to count number of signal within a single cell and then counting to have 100 cell, something like that. This is a normal. This is a normal. Let's say not normal. It is negative for alteration. So you have two somewhere two red dots, two green dots, and then in the majority of cell. I just connect to the question that one of you placed on the chat. That is, how are biomarkers and fish techniques applied in personalized medicine for cancer therapy? So, as I previously introduced a little bit with 1p19q deletion. So this is a diagnostic marker that it is very well used to diagnose oligodendroglioma instead of astrocytoma. And it makes difference in addressing the patient management. This is from a general point of view. To go deeper, we are testing with FISH HER2 amplification for breast cancer patients. If they are HER2 positive, oncologist can prescribe Herceptin, or which is the target therapy that is specific for HER2 positive patients. So this is for personalized medicine for treatment specifically. We have also I say diagnostic market that by changing the diagnosis, obviously as 1p19q for brain tumor, modify patient management and the protocol that it is used to treat patients. I mentioned HER2 amplification, so I try to connect all the dots, just not to go from a side to side without a logic. Those are two image pictures for HER2 amplification. The HER2 gene is labeled in red, the centromeric probe, which is the control probe, in green. So you have two different levels of amplification. And what I did, I start enumerating all these red dots. So let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and so on. So you have 15 red dots and 1 green dot in the second image where you count all the dots. Or you have cluster amplification. That means they are like, big bubbles of red signal, but all of those big bubbles are made of different dots. Okay, I've seen a lot of questions. Maybe I can address some of them later about machine learning, because I have some that. Advanced, I have also small of that. I can address the clinical utility of combining genetic biomarkers with genetic traditional diagnostic method. So let's go in that one. So this is a gene deletion, as I mentioned, 1p19q. So you have one red dot, two green dot. That means that one chromosome is deleted. In that case, for brain tumor, which is the algorithm basically, as many of the other situation, maybe probably the majority, there is an X-ray or imaging examination, and then there is a blood test to test all the standard biomarkers, and then let's say a biopsy. What we discover is that if it is in the brain, tumor of the brain, in that case, commonly oncologists match this data, so those data about 1p19q deletion, with the imaging. So if they see a tumor that sees in the temporal lobe of the brain that harbor 1p19q deletion, it is commonly more probable that it is a oligodendroglioma than an astrocytoma. If it is 1p19q deleted, it is for sure an oligodendroglioma. If it is on the frontal lobe, but it is deleted, that lesion may need an additional, set of analysis maybe mutation with NGS or with standard PCR and so on, because the position in the brain lobes given by the imaging is not fitting what is expected for an oligodendroglioma, which is commonly on the temporal lobe. So I guess it may address a little bit your question about the clinical utility of combining the multiple biomarkers. Again, for prostate tumors, you have specific rearrangements that are commonly used for to cluster the tumor in the different aggressiveness scale, treatment opportunity, and so on. But also you have PSA, which is prostate specific antigen, which is commonly used to monitor the post-surgery follow-up for patients. So that is a nice example of the combination. So we did fish, when I did fish during my experience in the lab where I working in, the fish is just a very important key data, but that need a whole set of anamnestic data or other, as I mentioned, imaging data, other biomarkers data like immunohistochemistry data, like histological data, like the number of, blood vessels per optical field, number of, the mitosis number, the Ki67 immunohistochemistry, positivity, that means if it is acting, replicating, and so on. Again, this is a better image. It doesn't suffer from the, another example, so from the translation into PDF. This is for fusion. So you have the normal tissue is that. So you have one red and one green, which is the normal, the control, or the two parts of the gene. And this is maybe BCR and able. This is BCR able translocated because the red and gene match. So the BCR and able that are far away commonly merge, and you can see the two probes that join together. And you can detect it, and you can see how many cells have this condition and where those cells are located, which is the shape of cell, and so on. For the break-apart probe, they are probes that detect when two signals breaks. That means there is a translocation within the gene we are mapping. A premise that I missed before introducing that one. We can use to detect BCR able, we can use or a fusion probe, so one probe on BCR, one probe on able. When the gene fusion occurs, probe are joined together, and you have that side. Or we can use a probe on, let's say, able, and when BCR join in, the probe split apart, and so you have one red and one green instead of one yellow. The different approach is when you have one well-known fusion partner, like BCR able, for instance. So you know that the clinical utility is just when BCR match able. So you can easily have a probe on BCR, a probe on able. When, for example, in lung cancer, just to keep the line with the example I already made, if you have a lung cancer sample and you are testing for ALK rearrangement, ALK, obviously 98% of rearrangement are with AMA. So there is ALK, AMA fused together, and you have the fusion, so, or ALK rearrangement. But if you just test for this specific fusion probe, you miss 2% of patients. That it is not a number, so it's not a human being, are not statistics, are singularity meshed together. So missing 2% of patient means that you miss 2 patients over 10 that can benefit on the treatment. So they design a probe that is on the centromeric side of the ALK, the other one on the telomeric side of the ALK. When one of the partner join in, the two probes split and you have that signal. Okay, let's go to some of your question and then. So I have a question to start. I'm not missing the one about the crisp and the other one. Just go to the one that are more in line with the part I already speak. So in terms of scalability of cost, fish as a routine biomarker validation, it depends. So scalability, the main bottleneck is in the microscopic analysis, the analysis at the microscope of the fish. The costs are obviously a little bit higher than a PCR, but lower than an NGS. And what can I say is that by using fish test, so the pain point is having a good biologist that can address your test. If you are testing a very high number of tests, probably it makes sense to have a automatic system that can acquire the image and then screening and scoring images for on behalf of the laboratories. But the biologist or the technician need to be here to check that the AI is making the right score and the right work. So this is the main bottleneck. Regarding the cost, probably you will repay the direct cost by speeding up the potential adoption and in writing down your, if it is a research project, your paper. Because having a test that is validate, having a test like the fish test that validate your biomarker means for the reviewer and for the clinic at the end of the day that you are using a very wide accepted gold standard technique to detect a biomarker with clinical implications. So it is easily to have this in the routine. So most of the labs already have NGS, but NGS provide clinicians too many information. For my opinion, let's say, they are overwhelmed by information or by mutation that make no sense from a clinical point of view. But the bottleneck is the people at the microscope that read the fish. And bottleneck in terms of scalability and cost. In terms of genetic biomarkers found in fish used to discover new drugs. So to speed up, so if you already know, for example, I give you a very, very practical example. I studied a lot on breast cancer. In the city where in my university, let's say, we had a very important breast unit for the region here, not as big as maybe you can have in your university, but we have a lot of male with breast cancer, which is a pretty rare disease. And we, just by connecting dots, we started thinking about how the androgen receptor gene and the on the X chromosome, obviously, if, let's say, the androgen receptor gene is involved in the male breast cancer, because, male is supposed to have one X chromosome only, but there are a certain type of male, especially those ones that have Klinefelter syndrome. They have two or more X chromosome that have the same incidence in breast cancer as female patients. So my first point is to start to see, okay, Klinefelter syndrome positive male have the same rate of breast cancer as female. What makes those two groups similar? The number of X chromosomes. So we started using a fish probe for AR, androgen receptor gene, and X chromosome centromeric region. And we screened a patient with gynecomastia without tumor. Patient with gynecomastia with tumors, male without history of neither gynecomastia or breast tumor, and patient with breast tumor without Klinefelter syndrome. So we discovered that statistically, and so this is a way that allow us to give a possibility to patient that got surgical removal of breast for gynecomastia to be tested for a marker that can allow them to have a shortened follow-up to prevent breast cancer, to occur in male. This is how fish can be used to discover new, new target. Also, androgen receptor, there are treatment that target specifically androgen receptor. So eventually, those group of patients can be specifically treated with androgen receptor inhibitors earlier to block tumor. This is a way. Obviously, you need to know the marker, and you can have custom fish probes to check that marker. Drug safety and effective. So it is more for effectiveness than for drug safety. You can use fish for drug safety because by checking centromeric probes, so the green probe, if the centromeric probe is scattered, so instead of having one green dot, you have multiple dot, especially in non-pathogenic cell, you may have a marker that it relates to the DNA damage. So you can say your treatment at a very low condition damage normal cells DNA, and it is bad, So this is for safety, but this is more for effectiveness. Alzheimer's and Parkinson's disease, it is a tricky point. There are several biomarkers less genetic, and I'm not an expert on neurodegenerative disorders, to be honest. What I'm would like to point about that is when we tested the conditions. So step back. All of those biomarkers are now tested because there is a treatment mainly available in the clinic usage. In the research use, they are tested because there are potential. So to discover a potential drive mutation or markers for condition that may have not right now a treatment, but for those conditions for whom it is possible to mutuate treatments from other conditions. Think about HER2 target therapy for ALK-resistant lung cancer that brings HER2 amplification in them. For those Alzheimer's or Parkinson, for R&D, it should be just for R&D. For clinic, I'm a little bit reluctant to put those markers in clinic because there is no treatment. So you can open up a Pandora pot of of stuff that may happen. And so think about if you say to someone that okay, you tested positive for a biomarker that it is strongly related to Alzheimer's disease to a 40 years old man. So what can you do? There is no treatment for Alzheimer's disease, just some action that someone can take, or for Parkinson's diseases. So how is how we will approach the future years having that big timing bomb in his body theoretically? So regarding the I have a question about machine learning, bioinformatics tool to improve validation. So this is the main, also the bottleneck and the pain point I always find when I'm doing demo. So apologize for the M that is not aligned with multiple cells. So sometimes before the modern AI, let's say, or modern machine learning tools, all of this scoring system is made by manually by operators. I also do manually by myself because if you have an operator with enough experience, that means 5 years of experience is just enough. They are faster and more reliable and more accurate than current AI system that make the same stuff. What we are developing right now is something that support operators is through machine learning and AI algorithm in identifying when the test fail. Because when the test is good, a good operator is very good in providing result. Sometimes I say better than a AI tool. But when the test fail like that one, you can see there are nuclei without dots, pretty dark blue, or with the holes inside. If the operator is not very well experienced, he may fail in identifying that there is a problem and where the problem is. So machine learning can a little bit annotate nuclei and say, okay, you can account the nuclei in yellow with a yellow shape because they are normal, but please be aware that the one with the red shape and the other one with no shape, something is not working, or I'm like a system I'm not able to detect where the nuclei board shape is, so you cannot count them. So machine learning and AI improve the identification of the, let's say, if you apply them from a statistical point of view, you can give them a lot of data, and as you know probably better than me in that case, and say, I have this protein, I identify this mutation in lung cancer. Please find statistically based on which other cancer types that mutation can be a driver of resistance, aggressiveness, or whatever. So we can starting study crafting a project, a specific R&D project to detect if that statistical based evidence is true, and to start treating those kind of different patients with that drug and allow them to have a treatment. In that way, it is a discovery phase, but also when you're going to, score biomarkers with fish, or eventually with immunohistochemistry, or so NGS is different because you already have an informatic data that it is, elaborated and managed by software. With the imaging, so immunohistochemistry, histological counter stains, or in situ hybridization, you can have image analyzers that with AI and the training that the important part is the training. So we have a library of 30,000 images, and we are going to train the tool that we are developing with those 30,000 annotated images to be able to provide the reliable data. But you need that amount of images. And regarding this CRISP-Cas, yeah, because I'm more familiar with NGS than CRISPR, but they are a way to detect mutations without knowing what mutation you are looking for. So just a first level of screening. So NGS to me is the most powerful tool right now because of the cost. So which is it is dropping down since it was discovered and in place. So if you have the possibility to say, okay, I have a very rare condition, which is that type of tumor A. I have 10 patients in my list. It is in that for rare disease, this may be a statistical relevant number. Let's go to a very deep whole genome screening of the tumor of those patients, of the normal tissue of those patients. You will discover hundreds of mutations. Then you can go deeper and see the, how the clinical side of those patients, so how they respond to treatments, if they are treated and so on. And then starting from the massive mountain of data given by NGS or CRISPR eventually, you need to dig into this big mountain to discover, to pick the just right one or two that can be relevant from a clinical impact. For R&D, it is so. If it is a project that it is for R&D only, you can just publish that you find 100 of biomarkers that are common in 100% of patients with the tumor A you are testing. For clinical impact, you need to go deeper and just dig just one, two, three markers eventually, or combination of them. And yeah, I guess I address all the question in that. Yeah, I already introduced that, but just to summarize some example of that, you have a case study. So, chronic myeloid leukemia, BCR-ABL as a drug target. Breast cancer, you have HER2 gene amplification that guide the target therapy and also is predictive of metastasis location. So, because if you have a patient with HER2 gene amplification, the target therapy is an antibody, which is specific for HER2 gene, HER2 receptor. Antibodies are not able to pass the brain barrier. So commonly those patients are more potentially developing brain metastasis. We are speaking about 20 years progression-free survival time. So after 20 years of the treatment, they potentially develop brain metastasis. Probably the fact that most of those patients are female that are more than 60 years old, that means that after 20 years, 25 years, they are 85. So they probably will never have any physiological impact if a metastasis occurs. Or they potentially have more bone metastasis in the hips and in their spinal cord, let's say. Or AML4 gene fusion that enable the new class of lung cancer drugs for non-small cell lung cancer patients, like crizotinib. So what we learned, I can pass to that. So pretty quick. So biomarkers can ensure patients receive therapy tailored to the tumor biology. Shortened the trials because you already started with a very, very well selected niche of patients, but also improve patient outcome. So CML, 10-year survival soared, as you see, from 30% to more than 85. That means that you, before having the BCR-ABL, 7 patients die, now maybe 1. So it isn't a life count, pretty important progression, improvement in life counts. Advantage of fish, I leave that slide for you to read, so I already told that. What I can stress a little bit of those slides is the high sensitivity and specificity. So when we design a probe for fish, if it is correctly designed, the specificity is almost 100% because it is a complementary DNA or nucleic acid fragment. And you can just detect if there is a single cell positive, you can detect the single cell positive. So you have also in fish testing on tissue slides, you always have positive, negative control, which is a normal cell, so or a fibroblast or a lymphocyte that is a control within. That means false positive and false negative are pretty low. And also fish, as mentioned, is not a substitute of immunohistochemistry and sequencing. As a startup as a startupper, let's say, former startupper, let's now try to call myself an entrepreneur also. In the diagnostic sector, I always not totally aligned with people that came out with new technique in diagnostic space and say, oh, my technique will replace immunohistochemistry, will replace sequencing, will replace whatever. Because in the clinical reality, there are diagnostic algorithms that are very well approved. And if those algorithms are still in place with immunohistochemistry, fish, and sequencing, despite all those technologies are pretty well mature, are pretty well available, that means that they provide clinicians with a different level of complementary informations that are important or key to have a proper patient management or a proper drug discovery process. So be always aware, and please don't tell that if you have a startup, that or from people that come into your lab and say, okay, I have this technology, we replace all the technology that are right now in place. But again, this is probably an unpopular opinion, but it is, I guess, more realistic than how it is supposed to be. Think about that. I started doing fish in 2003. So that not make me also elderly, but since I started, any year, someone from Thermo or Roche or one other of the Abbott or all the big Beckman and so on, jump into my lab and say, okay, next year we will come out with the NGS. It will replace fish test. The following year, NGS came out and the WHO added one marker by fish to the guideline. And it is always the same with the arrays TGH, Sanger sequencing, NGS, PCR, immunohistochemistry, artificial biomarker, and so on. Because fish is the tool that give you a visual confirmation, colored, that can prove that a mutation is in a specific cell with a specific relation of the surrounding tissue. Also, it is quick because in a couple of hours with the right tool, you can have a fish test. So theoretically, you can have a diagnostic test from a fine needle aspiration with patients already in the waiting room recovering for the fine needles aspiration. That you cannot have it with an NGS or with an immunohistochemistry. Maybe with an immunohistochemistry, but with an NGS, it's not possible. And by this speedness, you can have for your trial, you can have a rapidly, pivoting moment or not to go decision. Because if you see, okay, let's go deep to my example. We say, okay, and that project was part of my PhD, so I took care of that. Let's say we have this theory, crazy theory. So male with breast tumor, apologize. Male with breast tumor has, to me, more X chromosome. So the tumor of those male harbor more X chromosome that the normal tissue, and they look like, from a chromosomal point of view, to the female breast cancer. This is pretty, a kind of serendipity that we have. But if I have to go through an NGS, I would have spent probably 20,000, 40,000 dollars, US dollars in lab cost. I have to wait 1 month because you have to process 20,000. So we collected 40, 50 patients. That means 100 samples. So normal and tumor, gynecomastia, so more eventually. And potentially, if I failed my assumption, I wasted months and money. But what we did is to bring a probe already available for androgen receptor gene and the X chromosome centromeric region. We just cut slides, test 50 samples, 100 samples. That means probably a couple of weeks to me, because I'm pretty quick in doing, I was pretty quick in doing stuff. And in a couple of weeks, by spending for probably 3,000 euros, more or less. So in Italy, we have a very low, we are working with a very low budget, or we worked, or I worked with a very low budget for the PhD, also for myself. So pretty conservative in expended money. So 2 weeks, and I have my go to the project. I say, okay, I discovered that correlation. Let's go deeper into further study. So that means that in a couple of years, we published a lot on the male breast cancer androgen receptor gene and chromosome gene, X chromosome polysomy. So theoretically, we are now have enough evidence to go to a clinical study for that, but obviously it was for academic research only, unfortunately. And yeah, those are three key takeaway I would like to you to have. So precision medicine is here and will be more and more. Probably the time for individual medicine. That means that I have to be tested for X biomarkers. My brother can be tested for the same X biomarkers. If just one is different, a clinician must give us two different schedules for the same treatment. Is not. It was something that started when I was at the university, so in 2000 and early 2000. Is probably not a real possibility right now because it was mainly based on the cytochrome biomarker, liver cytochrome P150 protein, that are the protein that metabolize drugs, and they are very easily regulated by the environment. So if you test my activation, the activation of my cytochrome P450 today, that I had my training at the gym and I have my food supplement, my cytochrome is pretty high active. If you are going to test my cytochrome P450 tomorrow, that I will not go to train myself because I'm 40, so I cannot train myself 2 days in a row. My cytochrome activity will drop dramatically down. So it makes no sense. But precision medicine means that the treatment will be tailored to the tumor biology. This is the real precision medicine to me. And that means that in your discovery, you will need to find biomarkers that are really informative. So if you say, okay, I found a drug that P53 mutations, but P53 is not specific. So, or probably 90% of tumors have the P53 mutation. And this is not the right driver of the tumors. You need to target the right driver of the tumor and improve, obviously, the success rate of your trial. As I say, biomarkers make your trial faster. If you find the right technology or the right combination of technologies, you can speed up further. And always keep innovating. Probably I put it as the last, but probably it's the most important in this in this speech, because again, non-small cell lung cancer. We already have X antibodies for immunohistochemistry, probes for in situ hybridization, PCR, and so on that can test ALK rearrangement, but tumors are so the battle, the fight between tumor and drugs is adaptive. So sometimes at a certain point, it is possible to select a line a cell line within a tumor that can be resistant to the gold standard treatment for ALK rearrangement, like crizotinib. And if we stop to try discovering biomarkers in well-known conditions, we may miss the opportunity to treat patients that are resistant to that gold standard, and eventually for startup company opportunity to improve the startup business, let's say by saving life. We have now plenty of technologies that can support in doing that. First of all, AI, that to me is if well deployed and well used, can be a first brainstorming tool, and so then use it. uh, my last sentence that I want to close, because fish is something flourishing with light, so biomarkers light the way from discovery to cure. So thank you very much for your attention. I hope it was interesting to you. I will send to the organizer that group of slide, and any more question, happy to reply. You have my email. Feel free to email me, and yeah, I open the stage for to you.
Okay, thank you so much, sir, Dr. Indruga, sir, for that eye-opening presentation. Your insights on the role of genetic biomarkers and fish in accelerating drug discovery have abroadened our understanding of precision medicine. You talk highlighted not just the medicines or science, but also the transitional impact on patients' care and the cancer diagnostic. I'm sure our audience found the session extremely valuable. On the behalf of the organizers, we sincerely thank you, Dr. Indrugo sir, for sharing your expertise. We also thank you, our participants, for their engagement. Please stay with us as we continue with more exciting session ahead. Thank you.
Thank you. Thank you very much.
Shri,
Yeah.
there is a question from Dr. Ahmed.
It's me, Amara Aman.
Amra, yeah.
Yeah, it's me. Yeah, I have a question. Thank you so much for your time, sir. Can you please, if it's feasible, go back to ABL and BCR slide?
Okay. Let me share it again. Okay. That one?
Yes, that one. So, sir, basically, this slide represent a translocation between the ABL and BCR. So here my
Yeah.
question is practically what happened that leads to increase in the gene expression like after the translocation, although the gene are present in the first place as well. What are the factors
Okay.
that led to increase in the gene expression after translocation?
Yeah. No, very good questions. Thank you very much. So, this is I can address your question by giving also a general explanation. So, in translocations, they are relevant because commonly there is a constitutively expressed gene that share the promoter with a gene that it is supposed not to be constitutively expressed like ABL. So in that case, just because they are matched, the expression is driven by the BCR. So the BCR it is constitutively expressed. So that means that the protein that it is produced by the ABL, it is commonly it is massively produced, like ALK, MLF4 in lung cancer. So the way is the same. So when a translocation or a fusion, let's say in that case, drives the tumor grow, it is because the promoter of a constitutively expressed gene is used, let's say, to express a gene that commonly is highly regulated. This is the explanation. I don't know if it is clearer now.
Yeah, it's clear now. Thank you. I have a further question as well. Like here, you said that you are using fish. So by using fish, how can you assess or differentiate whether the gene expression comes from the gene amplification or the chromosomal duplication?
okay, like I can go to the picture, let's say, okay, this is an amplification because you have the green. So always in fish, you have a combination of probes. One that I call a test probe, that in that case in the red, which is the HER2 gene probe. And the other one, I call it control probe, which is the green probe, which is on the centromeric region of the chromosome that harbor the gene we are going to test. So take, for example, the cell that is that one. So you have two chromosomes because there are two centromeric regions that are highlighted, and those are several copies of the HER2 gene. This is a pure amplification because you have two chromosomes as in homozygosis and more than one gene. You may have polysomy. I don't think I have, my bad. Yes, there is one, that one. So the annotation is still there, but we can note on that. This is probably ALK or doesn't matter. So you have 3 control probe and 3 test probe. So that means that this is not an amplification, it is a polysomy of the chromosome that harbor those genes. This is the way we discriminate between poor genetic amplification or chromosome aneuploidy.
Okay, by using these different type of probes, right?
Yeah, by using basically a combination of probes that always have one probe for test, one for the chromosome enumerate. One is called the centromeric probe, commonly is called the chromosome enumerating probe, CIP. So having those two probes, the genetic locus specific for the gene and the chromosome enumerating probe, you always have a control over the polysomy instead of with the amplification.
Okay, thank you.
Thank you so much, sir.
Okay, thank you very much for your time and enjoy the rest of the speech.
Yes, sir. Sakshi?
Yes, are audible and
Yes.
okay. Good afternoon, everyone. I am Sakshi Shashikandabe and I am the honor to the anchor today poster presentation session at the third annual Swalife International Conference on the Innovation of the Drug Discovery. Through we are the virtually, their spirit to collaboration and curiosity is the very much alive. Today we are the exploring a diverse range of research through this visual storytelling is each poster window into the innovation and inquiry. Now our last poster presentation is a presenter is Amara Aman from the University of the Staffordshire. Here the poster title is Investigation of the Synergistic Effect of the capecitabine in the ion channel modulation on the cell proliferation, differentiation, cytotoxicity, and migration of their non-small lung cancer cell.
Yeah, here is me, Amara Aman. Good morning, everyone. I'm just going to share my screen. Yeah, here we go. So I'm Amara Aman. I have done my post-graduation from University of Staffordshire, and today I'm going to represent my MSc research project conduct under the supervision of Professor Dr. Ahmed Haidari. So basically, the title of my research project is investigating the synergistic effect of capecitabine and ion channel modulation on cell proliferation, differentiation, cytotoxicity, and migration in non-small cell lung cancer. So, to start with, cancer remains one of the biggest global health challenge despite decades of research and significant progress in diagnosis and treatment. It is a leading cause of death worldwide. And what are the factors, what are the aspects that make cancer particularly difficult to treat is its complexity. Complexity, like cancer cells, they have ability, they can rewire their metabolism, they have ability to evade immune system, and they can also, and they are so clever that always in response to any stress, they have ability to activate survival pathways, and they get successful in it as well, that allows them to resist the therapy. Another layer of complexity is metastasis. Metastasis, it is a process where cell undergoes epithelial to mesenchymal transition, known as EMT as well, that give them ability to migrate and invade distant organs. This is what make cancer so deadly. So, among different cancer types, lung cancer is the leading cause of cancer related deaths worldwide, accounting for more than breast cancer, colon cancer, and prostate cancer combined. And among lung cancer, 85% of the cases are non small cell lung cancers. Unfortunately, even though therapies have improved, many patients I'm unable to zoom it. I don't know why. Yeah, sorry for the disruption. So, among all cancer type, lung cancer is the leading cause of cancer-related death, accounting for more deaths than breast cancer, colon cancer, and prostate cancer combined. So, from the lung cancer, non-small cell lung cancer contribute 85% of the cases. Although there are several therapies and improved one as well, but many patients relapse because tumor become resistant to treatment. So, coming towards my research, in which I use an anti-cancer drug with an ion channel modulator to check either it increase the drug efficacy, either it gonna increase this combination gonna increase the effect of drug or not. So basically, I use capecitabine, which is a chemotherapy drug and is commonly used against solid tumors. However, its success in non-small cell lung cancer is limited due to dose dependent cytotoxicity and frequent emergence of drug resistance. So, on the other hand, ion channel blockers, these drugs usually given for the heart rhythms problem, but they are increasingly being linked to cancer cell migration and invasiveness. And their increasing trend led us to an important question: could combining capecitabine, which is an anti-cancer drug, to an ion channel blocker create a stronger anti-cancer effect by targeting both cancer metabolism and electrical signaling that drive cell invasion? So we plot our research on this question. Coming toward the aims and objectives, the aims and objective of my research is to investigate the synergistic anti-cancer effect of capecitabine and ion channel blocker in A549 non-small cell lung cancer by evaluating their impact on metabolic activity, apoptosis, morphological challenges, and expression of genes related to EMT and cell cycle regulation. Moving towards the material and methods, Here is the overview of experimental design, which systematically tested both drugs at two concentrations individually, means the ion channel blocker and the anti-cancer drug individually, and their combination, a lower dose combination and a higher dose combination. All experiments were conducted in triplicates to ensure the reliability of the experiment. Several lab approaches were used, like MTT assay to check the metabolic activity of cells, flow cytometry to check the apoptosis, fluorescence microscopy to evaluate the morphological changes after the treatment, and then RT-qPCR to analyze the gene expression analysis. After that, ANOVA analysis was used to check the significance of the results. Coming toward the result, first of all, the MTT assay was performed. As I mentioned earlier, it was performed to check the metabolic activity of the cells. We set up our experiment in two sets, one for the 24-hour and the other one for the 48-hour, and the results revealed a time and dose dependent reduction in cell viability for both ion channel blocker and the capsaicin. Importantly, the combination groups demonstrate greater inhibition than either drug alone, and this strongly indicate a synergistic cytotoxic effect, particularly at higher doses. These are the results from MTT assay. Furthermore, MTT assay followed by the flow cytometry. Flow cytometry basically confirmed the findings from the MTT assay, like at 24-hour we observed reduced viability with both drugs, but the strongest reduction occurred in the combination groups. By 48-hour, the higher dose combination yielded the lowest survival fraction, validating the synergistic cytotoxicity seen in the MTT assay. Sorry?
ऐक जरा.
What happened? I'm unable to zoom the screen. Okay. They are going to zoom. Okay.
5 minutes left.
Actually, I'm unable to zoom my screen, but I want to zoom my result section. So, can you please help me to do it?
Yes.
Camera, you just come out from the sharing. I will share from my side.
Okay. Yeah, I'm done.
Yeah, can you see?
Yes.
Okay.
So here I was talking about the flow cytometry. That flow cytometry, the cell viability reduce in a time and dose dependent manner. Like in 24-hour, the control has viability 90%, but when it comes towards the drug implication or the ion channel blocker implication, the viability reduce. Moving towards the last, the combinations at the sir, can you please move a bit down? Yeah, here, a bit up at flow cytometry section. Yes. And when it comes to combination, the cellular viability reduce up to 66%, like control holding viability up to 90%. But when it comes to the combination, the higher dose combination, the viability reduces up to 66%. Same happen in the 48-hour as well, the control holding 92% viability. But when it comes to the combination, the viability reduce up to 55%. So it strongly suggests that the combinations, they are working synergistically, means they are validating the synergistic cytotoxicity as seen in the MTT assay as well. Moving towards the fluorescence microscopy, here, yeah, fluorescence microscopy, here using acridine orange fluorescence we visualize apoptotic morphological changes, including nuclear condensation and membrane blabbing as well. Here in the control, the cells, they are quite adherent. They are present in the form of groups. But when we move towards the our treatments, the apoptotic features, they get pronounced. Like in the combination groups, supporting the idea that maxilithin enhances the capecitabine cytotoxic effect. In the next figure, we notice that cell becomes shrink. Like in stress condition, I took this picture in a zooming view. In the stress condition, cell tries to communicate with each other. And here we may said that there is an electrical signaling communication between the cells. It is interesting to consider that cancer cells often try to communicate or adapt different survival pathways under stress to find survival or escaping routes. So, what we are seeing here may reflect the blocking of such communication pathways, leaving the cells more vulnerable to apoptosis. Due to the short 6-week project time frame, I was unable to investigate this phenomena in depth. However, it is definitely promising observation that deserve further exploration in further studies. Moving towards the next section, RT-qPCR revealed significant findings as well. Like E-cadherin gene expression was upregulated. E-cadherin is an epithelial marker, so its expression get upregulated after we apply treatment to our panels. Like in the control here, see a significant upregulation comparing to the control, and moving towards the combination, it suggests EMT reversal. So, moreover, the next one is the MMP9. MMP9, it's a mesenchymal marker. It shows a reduction as well. So these basically reduction in metastatic potential. This provide molecular evidence for anti-metastatic effect of drug combination. Moving towards the discussion, these results suggest that combining capecitabine with ion channel blocker not only enhance cytotoxicity but also reprogram cell at the molecular level to make them less invasive. This dual action could help overcome drug resistance in non-small cell lung cancer. In summary, this study basically highlight a novel therapeutic approach, repurposing ion channel blocker to strengthen chemotherapy in lung cancer by targeting both survival and invasion pathways. This strategy may provide a promising way forward for patients with limited approaches. At the end, I would like to thanks to my supervisor, my technical team, and University of Staffordshire for providing me the platform and also their help and guidance. Thank you so much. If you have any question, you can ask.
Thank you so much for that inside the presentation. Some question for the audience. First question is how do your specific ion channel contributed to the synergistic cytotoxic observed with the capecitabine?
Basically, these ion channels, they help to reduce the cancer invasiveness. Means the ion channels basically what they do different. Like if I talk about the sodium ion channel, the blockage of sodium ion channels help to reduce the metastasis. Mean it restrict the cancer cell from invasion. So this is how they are not directly contributing, like they are not directly helping the capecitabine. Like capecitabine, how capecitabine work, it damage the DNA, it impair the cell cycle or the regulation of gene that contribute to the growth. But ion channels, how does they work, they create that type of environment and they promote the effect of drug. Like they change the pH level or things like that. In this way they're gonna help the anti-cancer drug rather than involving in the molecular mechanism.
Okay. Our next question is what is the role do the ion channel play in the cancer cell behavior, particularly in lungs cancer?
As I mentioned earlier, the ion channel basically we can take. I in my research I took the sodium ion channel, and this ion channel help cancer cell it restrict the metastasis of the cancer cells, like it stop the invasiveness by yes, it stop the cell invasiveness, and by altering the pH of the environment. It also helps cancer cell. What they do, basically they start degrading their surrounding environment, they start degrading the matrix to make a space for them, and these ion channels stop them by doing this.
Okay. Our next question is what is the rationale for the combining capecitabine with the ion channel modulator in cancer therapy?
The rationale is basically when the capecitabine is the rationale of the study is basically when the cancer drug is using alone, we have to use the different doses of the drug. Like we start from the lower dose to moving towards the higher dose of the drug. So when we are applying lower dose of drug to the cancer cell or to the cancer patients, the cancer cells, they become resistant to that dose, and even at a particular time frame, the higher dose didn't show any effect. So the rationale of study is what we are going to do, like we are using ion channel blockers, so that even the lower dose become more effective. As it's obvious in my results, in my findings as well, that when we are combining the capecitabine with ion channel blockers, the cellular viability reduced significantly as compared to the drug alone. So this is the rationale of the study.
Okay.
Yeah.
If any questions? No.
Hello. Yeah, I got one question.
Yes.
Hi, Amara. Nice
Hi.
presentation. Just one question regarding the MTT assay you have done.
Yes.
The cell line which you have taken, both are cancer cell line, right?
Yes.
So have you tried testing on normal cell line?
Basically not. These normal cell lines was not provided. We get only the cancer cell line from our academic teams. Even it was same thought I have in my mind as well. Like we are comparing the cancer cells with the cancer cells, because in normally cancer cells, if we compare them with the normal cells, they have different metabolism. Even we didn't if we didn't apply any of the treatment or any of the stress to the cancer cell, they still try to change their shape. They still deviate. Their metabolism is very different from the normal cells. So it will be very good if we have a control as a normal cell, as a cancer cell, and then treatment panels. But in my research, we get only the cancerous cell lines.
Okay, and to show the effect of the migration, what assay you have done?
To show the effect of
Migration, cytotoxicity and migration you are doing, right?
Yes.
So which assay you have done in migration?
The particularly I have done these assays I have present to you: MTT, flow cytometry, RTQ, PCR, and fluorescence microscopy. So basically, particularly for cellular migration and for the apoptosis, we use flow cytometry.
Okay, so you have used flow cytometry. So you have not used scratch assay?
Sorry, what?
Scratch assay?
No, we didn't use scratch assay because we one of the major limitation of this project was the time. We get only the 6 week time. That's the reason.
Okay, in the 6 weeks you have done this.
Yes, in the
Yeah.
6 week time.
Okay, good, nice.
Yeah.
Thank you for your presentation.
Okay, thank you.
Thank you so much, Amara Aman. Then now I would like to invite my colleague Sanjana to the next comparing session further.
Okay.
Sanjana?
Yeah, am I audible?
Yes.
Good evening, everyone. As we come to the close of day one of the third annual Swalife International Research Conference on Innovation in Drug Discovery, it is my privilege to deliver the vote of thanks on behalf of the organizing committee. First and foremost, I would like to express our deepest gratitude to all our distinguished speakers for sharing their valuable knowledge and insight with us today. From Dr. Praveen Bade sir, enlightening talk on professionalized herbal formulation in cancer prevention, to the inspiring research presentation by our participants, and finally, the thought provoking session by Dr. Enrico D. Otto on generic biomarkers and fish. Each session has truly enriched an understanding of modern drug discovery. A heartful thank goes to the organizer, Swalife Foundation, SSTSI, and Swalife Biotech Limited for their constant support and guidance in making the event possible. I would also like to sincerely thank all the participants, researchers, and students who joined us today through Zoom and YouTube Live. Your active involvement, enthusiasm, and curiosity have made today's session interactive and meaningful. Finally, a special note of appreciation to my co-anchors Shreyash, the technical support team, and everyone working tirelessly behind the scene to ensure the smooth execution of today's program. With that, we conclude the first day of our conference. We are looking forward to welcoming you all tomorrow with yet another day filled with inspiring talks and innovative research discussion. Thank you everyone and have a wonderful evening.
Thank you, everyone.
Thank you all.
Thank you, sir.